for Professor Raiola, and within 1 minute we should start according to the program, okay? And you, okay. Okay, all the people are joining again the meeting, but I cannot see Alessandro Raiola yet. You are muted. You should be able to activate your, you are allowed to do it, Zlatko. Okay.
Sorry, Alessandro. Now it's all okay. Sorry about
yeah,
this.
it's okay.
It's some technical issues.
Okay.
Buongiorno, buongiorno,
Buongiorno.
from Zagabi.
So we're just waiting for the president of the commission.
No worries. What was the issue? Because I couldn't connect.
Oh, I made an error. I put in within the settings, I put that you should be you should have an account, but in this case, the account should be by my university, okay? And so it was made that there was a restriction to the people who were allowed to participate.
Okay.
It was a mistake of mine.
No worries.
Sorry
no
again.
worries. No worries. Just that it's good that we sorted it out, so it's okay.
Just another thing, I want to say that the cadeau is here in my office or in the Franco Major office.
Yes.
So if you are here, you can go here at the end of your presentation.
Yes, all the students. There was a message to all the students. I don't know why Raiola is not joining. Dove è finito? Please take patience. Be patient, take patience.
Cercando di guardare video su YouTube, ma non si sente niente.
Sì, sto cambiando il link adesso. Dite che sto cambiando il link di YouTube adesso. Portate pazienza perché ha creato un nuovo link quando ho reimpostato la chat. Ecco, se... Okay, I made a new YouTube, so please go to the web page of the website of the PhD in crop science, the link that you should have. There you can find the new YouTube streaming, okay? So the streaming has changed. So tell the people who are following you that there is a new link, and they can find the new link in the web page address that I sent you, that was sent you before by Arcangela. Go there, you should be able to find it. Let me know if there is problem, but I see it. Alessandro, dove sei finito? Oh, eccolo qua. Se non lo vedono, ditegli di aggiornare la pagina YouTube del dottorato, ok? Per il YouTube, per il link YouTube. Perché io adesso... Sì, dimmi.
No, gli dice che non si sente niente su YouTube.
Va a vedere adesso, Claudio. Bisogna aggiornare un paio di volte perché ci mette un po'.
Ok.
Riandate però non sullo stesso link. Tornate sulla pagina del sito del dottorato perché lì c'è il link nuovo.
Ok.
Adesso vedo me stesso, per cui ci dovrebbe essere. Ok, so we are Alessandro Raiola, ci sei?
Sì, sì.
Okay, so the three members of the commission are here, and I think that we have just 5 minutes of delay. And I think that, Alessandro, do you want to introduce the first student or?
Okay, if we are ready, we can start. The first student on the base of the program is Monica Canton, that will present us the regulatory mechanism controlling flower bud dormancy in peach.
Monica, you should be able to share your screen if you want.
Okay, thank you.
Okay, we see your screen.
Okay,
You can start when you want.
okay. So I ask maybe for my colleagues to turn off the microphone to.
Yeah, you should turn, all of you should turn off the microphone.
Yeah, thank you.
Claudio, anche tu spegni il microfono, per favore, così Monica si concentra.
Good morning, everyone. So my name is Monica, and today I will speak about my PhD thesis entitled The Regulatory Mechanisms Controlling Flower Bud Dormancy in Peach. And my supervisors were Claudio Bonghi and Serena Varotto. So,
Monica, just a brief, sorry, when there will be 5 minutes left, we will let you know.
Okay, thank you, thank you. I will start speaking about a short general introduction, then an in silico meta-analysis. Then I will show you bud dormancy characterization and the RNA-seq analysis, the DNA methylation and ChIP-seq analysis, and to finish, of course, the main conclusions. So the peach belongs to Rosacea family, genus Prunus. It's very used like a model plant, mainly in studies on genetics. And a very important characteristic is a temperate tree, which means grow during favorable seasons and goes to dormancy during unfavorable condition. Dormancy can be defined the inability to initiate growth from meristems under favorable conditions. And during this dormancy, the plant protects their meristems inside the structures named buds. In peach, we have a flower bud divided from a vegetative bud. So during this period of winter, all four vegetative organ verticiles, sorry, like here in this picture, so sepals, petals, anthers, and the carpel are all already present, but of course underdeveloped. So in this period of endodormancy is when the plant, the dormancy is controlled by internal factors like genetic factors and hormones. And then when the plant fulfilled his chilling requirement, the plant enter in ecodormancy. Ecodormancy is controlled mainly by external factors like temperature and photoperiod, until, of course, the bloom. So the main objective of this thesis was to elucidate the regulatory mechanisms involved in flower bud dormancy. Especially, was to perform a meta-analysis to understand the timing and the progression of bud dormancy in 3 different species: sweet cherry, apricot, and peach. was also to investigate the dynamic of transcriptome landscape by using the next generation sequencing in two nectarine genotypes, Fantasia and slow ripening mutant. In the same plant material, we analyze also the DNA methylation status. And of course, also was the objective to perform a chromatin immunoprecipitation analysis across the entire genome of Fantasia. So the meta-analysis, so we are an in silico analysis. We downloaded 3 RNA-seq datasets, so in peach, apricot, and sweet cherry. For each one, we choose a high and low chilling requirement. Chilling requirement means the amount of chilling necessary to break up the dormancy, break up the endodormancy, in order to specifically understand the dormancy in flower buds. So for this analysis, we concentrate our attention, especially in endo and ectodormancy. We were able to identify 5 main pathways involved in dormancy, in bud dormancy. And for each one of these main pathways, we are able to identify very good markers for each phase. For example, in endodormancy, the DAM genes, I will show you before and explain about these genes. DAM genes are very good markers to this phase for endodormancy. In turn, for ectodormancy, we are able to identify a very good dormance for macro and microsporogenesis. So the meta-analysis indicate to us that in ectodormancy, buds are ready to gamete formation because we are able to identify that genes of microsporogenesis and microsporogenesis. Markers of dormancy progression between these two phases were identified, since boundary between endo and echo cannot be easily defined. There is not a real dormancy in flower buds, and dormancy control involve more than a single pathway. Knowing all this information about this meta-analysis, we tried to transfer this knowledge to our experimental in Fantasia and SR. So we choose these 2 genotypes, Fantasia, the wild type, to study the bud dormancy. In addition, we also add this SR mutant. SR means slow ripening, in which previous studies showed to us that SR are incapable to produce ethylene, and also the firmness remain always very high, and the lignin content always very high. So probably these alteration in the SR fruits are before the fruit formation. So it's probably that this alteration is during the bud formation. So we add this SR mutant to support our studies on dormancy. So we collected the samples from September until January to preliminary RET analysis. Then we selected some of them to perform the RNA-seq analysis, hormone determination, and the DNA methylation. We select again two cytological observations, ChIP-seq analysis, and in situ hybridization. We collected these buds following a Utah model based on chilling units. Is a model to calculate the chilling necessary to break up the dormancy. And so in the cytological observations, we are able to identify, for example, here we have the 4 time points, the bud section, and following the bud dormancy progression, of course. And in the last time point, when the chilling requirement was fulfilled, we can observe the microspore mother cell. And this result, it's very a good result because it's in agreement with the meta-analysis results, in which gene markers expressed in ectodormancy. And similar observations are obtained in Fantasia. At the hormone level, so we performed the hormone quantification in collaboration with the University of Valencia. Here we have abscisic acid and gibberellic acid. They are the two main hormones responsible for dormancy. So abscisic acid decrease following the dormancy, and the gibberellic acid increase. There is no difference, there is no a big difference between the two genotypes. And at the genetic level, here we have the DAM genes, dormance-associated genes. These genes are until now the major regulators of dormancy. They are 6 DAM genes. I will show you just 2 of them, so DAM6 and DAM4. We observe an increase of these DAM genes during dormancy, and they decrease with the dormancy progression. There is no difference between the two genotypes. Sorry. We didn't have any information about the localization of these genes, so we performed on DAM4. We performed an in situ hybridization. So the purple color indicate the localization, the expression localization of this gene. It's a very general expression, so probably these DAM genes shall coordinate the dormancy. So these objectives, they coordinate all tissues together. Knowing all this information about the characterization, we performed a RNA-seq analysis. So to identify the differential expressed genes, we used 4 different approaches. So we are able to identify the differential expressed genes in Fantasia, in SR, between the 2 genotypes, and also to identify the code effect. So for example, here in Fantasia, we performed a gene set enrichment analysis to identify the main pathways involved in this process of dormancy. So for example, we identify flower oil development, response to temperature stimulus, negative regulation of flower development, and pollen exine formation. Very similar pathways, GO terms, were identified in SR genotype. But when we compare the 2 genotypes, Fantasia against SR, for example, in this heat map, we can observe 2 main clusters, and they show opposite trend, which means when in SR the genes are upregulated, they are repressed in Fantasia. And when they are repressed in SR, they are active in Fantasia. To identify better which genes are inside of this heat map and this function, and their function, we used a general molecular model to study these genes. So we have, for example, the photoperiod and the cold temperatures increase the abscisic acid. In turn, they inhibit a PICOL. PICOL is a gene responsible for chromatin remodeling. Consequently, SVP, there is an important gene, and here we have also the DAM genes, is a very similar function, increase also the callose production. Callose is necessary to closure the channels between the cells, inhibit also gibberellic acid, and consequently the plant the bud enter in dormancy. For example, so for the hormones, we have here in this heat map, in red we have high expression, in blue we have low expression between the 2 genotypes. And we can observe for example, abscisic acid synthesis, gibberellic synthesis, and also abscisic acid catabolism, there is a very difference between the 2 genotypes. And probably this alteration between the 2 genotypes is a time shift. In addition to the hormones, the DAM genes and also SVP do not have any difference between the 2 genotypes. They are very high at the beginning of dormancy, and they decrease with the cold exposure. But for example, in the callose deposition, we observe the callose deposition and callose degradation only in Fantasia. So SR again might be a belated development in confront of Fantasia. And all these differences, and in general also, not only these genes that I showed to you, but in general, differences may be due an epigenetic effect. For this reason, we performed a DNA methylation analysis. So we use 3 different methylation contexts. So in CG, CHG, and CHH, H can be a cytosine, thymine, or adenine. This DNA methylation analysis was performed in collaboration with Giampiero Marconi from University of Perugia. And we were able to identify the differential methylated regions. Basically, there is no difference between the 2 genotypes, but when we associate these differential methylated regions to the differential expressed genes, we observe that in SR has a higher number in compared to Fantasia. Here I show you at the CG methylation context, only Fantasia, because in SR we are still working the data. So in Fantasia, this relative methylation level, we observe a decrease during the cold exposure. To better understand, so we divided these differential methylated regions, we divided in upstream gene body and downstream from the transcription start site. We focus our attention only in the upstream genes because they are noted to repress the transcription, the gene transcription. And some of them have a very good correlation between this DNA methylation and the differential expressed genes. In this moment, for this presentation, we didn't have time to explain better, so I'm so sorry for this. In addition to the methylation, we have also the chromatin remodeling that is very important and influence the gene expression. So we perform the chromatin immunoprecipitation analysis. We choose 2 histone modification. For example, H3K4 3-methylation is associated to active the expression, and H3K27 3-methylation to repress the expression.
5 minutes, Monica. Okay.
Thank you. So for example, a genome-wide distribution of these 2 histone modifications, H3K4 3-methylation is across the TSS, the transcription start site. In turn, the H3K27 3-methylation is the entire gene. Both behavior is like the expected. To enter in detail, so we try to compare this enrichment, for example, here to H3K4, enrichment against the differential expressed genes. And we observe, for example, here in the dark green, we have the expressed genes. So when the genes are enriched in H3K4, most of them are also expressed. But when they are not enriched in this H3K4, for this histone modification, they are also not expressed, indicating that an increase of gene expression correlates with a progressive increase of H3K4 3-methylation. Here we have a screenshot, a genome viewer of, for example, DAM3, and we observe when the H3K4 decrease also the gene expression, so it's a very good relationship. But when, for example, here, gibberellic acid biosynthesis, for H3K27, when this histone modification decrease, the expression, the RNA-seq peaks here represented, increase. This gene I already showed to you before, and it's a very example of a gene controlled by environmental, DAM genes, and histone marks. In conclusion, so DAM genes, dormance-associated genes, are the major regulators, but only in endodormance phase. Although the fulfillment of chilling requirement is not marked by the completely zeroing of the DAM expression, flower bud differentiation slows until the end of endodormancy. The transition to echo is marked by the initiation of micro and macrosporogenesis. Differential expressed genes strictly associated with dormancy can result from DNA methylation and the deposition of these two different histone modifications. And during the dormancy, differences in the flower bud transcriptome landscape in Fantasia can be associated with DNA methylation and the deposition of these H3K4 and the H3K27 trimethylation. Some future perspectives, so studies on vegetative buds are necessary to understand the connection with flower bud development, especially on dormancy release. Identifying the exact environmental signal that triggers flower induction could possibly lead strategies to manipulate the bloom time. And the localization SVP expression could be useful to better understand the dormancy and identify the epigenetic markers mechanisms involved in SR are necessary. Thank you for your attention.
Okay, thank you, Monica.
Thank you, Monica.
Thank you.
There is questions, Zlatko, you have some questions about it?
Thank you very much, Monica, for your presentation. I have no additional comments at this moment because it was a little bit difficult for me to follow the causal relationships within the studies. I was just a little bit, I didn't understand a relationship between these chilling requirements and fruit ripening. Why was it mentioned at one point in the presentation?
Yeah, sorry.
I didn't understand why pointing that out, you know.
In this specifically slide, maybe?
Yeah.
Yeah, so chilling requirement, I mean, our objective was to study the dormancy. For this reason, we choose the buds, specifically flower buds.
So just flower bud, wild type without the flower. Okay.
Yeah,
okay.
yeah,
That's
just
cool.
flower buds. Yeah, and here I just contextualize the reason to choose SR, this mutant, because this mutant has a big problem with maturation. But here the objective is not to understand the problematic of this mutant, but so previous studies showed that probably the problematic of this mutant is before fruit beginning. So maybe in the buds. For this reason,
no, Monica, it's all good. It's just
okay.
I just wanted to make a point that it was a little bit difficult for me to follow without introduction into the topic a little bit more specific. Okay. But thank you very much.
You're welcome.
Thank you very much.
Other question?
I have several questions actually.
Whoa.
But I will just actually not real questions, but most of them are just curiosities and details that, of course, you couldn't tell us that because of the time. Okay, you couldn't told us because of time. So, in general, my impression is really positive. I mean, you gave us there are a lot of information in your thesis, and maybe too many informations, and it's sometimes difficult to have all the information in front of you to make connection and links and to understand the whole logical sequence of events, let's say. Okay. So, but I think that in general, what you showed, I think that I was thinking about that it would be interesting to compare peach with other crops, first of all. Just, well, I mean, to see if the mechanism that you observed in peach are conserved and at what extent they are conserved in other crops. The first that I think about is grape, for example, that I know much
more or
more.
less like we show in the meta-analysis, because in the meta-analysis we use
but I
different-
was talking about also the other part of your
Okay.
data. Okay.
Okay.
And some specific question concerning the meta-analysis. You know that when you make a meta-analysis, the first important thing to do is to normalize the data coming from different experiments.
Yeah.
Otherwise, you cannot compare them. I didn't find any normalization procedure in your materials and method, but I think that you did it. Did you?
Yeah, of course. We use a rescale from RPKM.
Okay, so just a rescaling.
Yeah.
Okay.
So we calculate the RPKM.
Yeah, I know. I understand that maybe it's not the best method, but anyway, you did it. Then another curiosity: why you didn't consider ethylene among the hormones involved in bud dormancy release? I'm asking you this question because, for example, in grape, it seems to have a clear positive role in dormancy.
Okay.
I mean, there are some experiments in which they treated the buds with they treated the grapes with ethylene, and they observed a significantly increased bud break. So I was wondering why you didn't consider ethylene in your analysis.
Well, based on the literature, in rosacea species, in very general model, ethylene don't seems to be a very important gene. Of course, there is a very good importance, but mainly on abscisic acid and gibberellic acid. I don't know why not is the importance. I mean, it's not so important. So like abscisic acid and gibberellic acid, but in general, they not speak so much about ethylene.
Okay, and concerning this, your second chapter, and concerning the gibberellins, I saw that you quantified only GA4 and GA1. Was it just for a methodological limitation, or you chose to quantify just those two gibberellins and not, for example, GA3, which is known to have a role in dormancy in peach, especially, in GA7?
No, because in this collaboration with the University of Valencia, they performed only GA1 and GA4.
Okay, so it was a methodological.
Methodological,
I was
yes.
already imagining that it was the problem.
Yeah.
Another curiosity about the methylation analysis.
Yes.
What I was imagining, but while I was reading your thesis, I expected that at a certain time you should have made a general correlation analysis between DNA methylation pattern and gene expression pattern. But you just made a target correlation. You chose just subsets of genes and so on. But I was expecting you to make a general correlation just to see if the methylation context does really have a certain correlation with the expression pattern, because this is always a problem. This is a problem that I encountered in some past analysis, and there is always a big question point on this topic. So I don't know if you can tell us something more about it.
So we expected also the same thing, but unfortunately, we are belated with the DNA methylation analysis, but we are in progress. And from something more months, we are publish our article on this methodology. But
you have in mind to do it.
Yes, of course.
Of course.
Yeah,
nice.
because it's very important. Yeah.
Please let me know the results. I'm curious. And last, your last chapter, but I think that the reason is the same that you told us. Maybe you are doing these analyses are in progress. I was wondering why you abandoned the idea of comparing Fantasia and SLR also in terms of histone marks.
Because it's ChIP-seq. It's a very little bit complicated analysis, and there is no reason try to understand histone marker in a mutant before understand in the wild type.
Okay, so it's the following step.
We start also before understand Fantasia and then making in the SR mutant. Also because for chip psychoanalysis, we need a lot of plant material and the buds are very small. So before we need to understand Fantasia and then perform it in SR.
Okay. Thank you very much, Monica.
You're welcome.
And congratulations.
Thank you.
Okay, I don't know if there is another possibility, but just I was wondering, one curiosity, on the basis of these studies, considering the fulfilling of chilling requirements and the adverse effect in climate warming scenario, about your opinion, what molecular markers can be adopted to select the best adapted cultivars in these future scenarios?
Well, here we don't select any marker to this scenario, this climate change. But of course, all these general identification of the genes are very important to the I mean, like can I say, to the genetic basis studies to perform, for example, the breeding or.
For example, there is a marker genes better characterized during ecodormancies to resume bud growth of some fruits.
Yes.
There is that refer to a specific hormone.
No, they are not. For example, here, yeah. For example, can you see my slide? My. So here, for example, NSEED5, it's a very good marker, and it's responsible to abscisic acid biosynthesis, and it's a very good marker to endodormancy, for example. And the same gene, but for abscisic acid decrease, a very good marker to ecodormancy. So it's important to know the genes that are marker each phase. And this probably it's very useful for the breeders, and they can produce new cultivars.
In this is not included brassinosteroids because I have read something about it.
No, for here, no.
Okay, thank you.
You're welcome.
I think we have to move to the next student. So please, Monica, unshare your screen. And thank you again.
Thank you.
And Alessandro, if you want to introduce the next one.
The next is Manuel Ferrari that will present the topic on sustainable agriculture practices to improve nutrition, grain yield, and quality in cereal crops.
Yes, good morning. Can you hear me?
Manuel, I will let you know when you have 5 minutes left, okay? So
Okay.
you can start when you want.
Can you hear me? Yes?
Yes, yeah. Clearly.
Okay. So, good morning to everyone. I'm Manuel Ferrari and my supervisor is Professor Teofilo Vamerali. My PhD project regards the evaluation of sustainable agricultural practices to improve nutrition, grain yield, and quality in cereal crops. In the last 60 years, the amount of nitrogen fertilizers in the world was grown by 90%. This large increase of nitrogen inputs has led to a progressive reduction of the nitrogen use efficiency, which is defined as the ratio between the kilograms of grain yield obtained for the kilograms of the total nitrogen inputs applied. Additionally, this large surplus of nitrogen may lead to damages to the environment and problems for the human health. For this reason, the research in agriculture is trying to find the best strategies in order to improve the nutrient uptake efficiency, reducing the chemical inputs through a better optimization of nitrogen fertilization. The main strategies regard the enhancement of use efficiency of fertilizers through the use of biofertilizers, the adoption of resource conservation practices, the improvement of the management and application of nitrogen sources, and foliar nitrogen spraying can be a good option, and the capability to determine the nitrogen status at soil, leaf, or canopy level through a wide range of sensors. For this reason, the aim of my thesis was to evaluate several agricultural strategies in serious cultivation in order to enhance the environmental sustainability without affecting productivity and grain quality. In the first work, we have evaluated the effects of seed-applied biofertilizer in wheat. With the term biofertilizer, we refer to a substance which contains living microorganisms as plant growth promoting rhizobacteria and arbuscular mycorrhizal fungi, which, when applied to seed plant surfaces or soil, colonize the rhizosphere or the internal plant tissues, promoting growth by increasing the supply or availability of primary nutrients. In particular, the plant growth promoting rhizobacteria can enhance the plant growth through the biological fixation of nitrogen or through the improving of the solubilization of low mobile nutrients. In particular, nitrogen fixation can be carried out by several bacteria that are mainly divided in two categories, symbiotic and non-symbiotic bacteria, while there are other bacteria known for improving the solubilization of other nutrients. It's the case of Bacillus megatherium, that is widely known for improving the availability of phosphate, and Frauteria aurantia, that is known for improving the solubilization of potassium. As regards the arbuscular mycorrhizal fungi, they have a lot of benefits for the plants. In particular, they enhance the water and nutrients availability, and the most known is the Rhizophagus irregularis. For this reason, we have evaluated the effects of 3 commercial biofertilizers products consisting in PGPRs alone or in association with AMF on growth, grain yield, and quality in wheat. The 3 biofertilizers were triple N, containing the 3 bacteria Azorhizobium, Azoarchus, and Azospirillum, Rhizosum N, containing the AMF Rhizophagus irregularis and Azobacter vinelandi, and Rhizosum PK, containing the 2 bacteria known for improving the availability of phosphate and potassium, Bacillus megatherium and Frauteria aurantia, and the AMF Rhizophagus irregularis. These 3 products were applied to the seeds at sowing time, and these 3 products were compared to an untreated control. The experiment was set up in open field at the experimental farm of the university, and Bologna was the wheat cultivar used for this experiment. During stem elongation and heading stages, the shoot dry weight and the content of nitrogen and of 6 nutrients were assessed, and at harvest, the productivity and the grain quality were the main parameters investigated. Going through the results, the most important results at stem elongation was found for the nitrogen content, where all the 3 biofertilizers led to a significant improvement compared to the control, and this result was due to an increase of the shoot dry weight and to an increase of the nitrogen concentration. At heading, all the biofertilizers increased the content of nitrogen and the dry weight, but only Rhizosum PK was statistically different compared to the control. At heading stage, all the biofertilizers led to a reduction to the concentration of the 6 nutrients, but in the case of calcium, potassium, and zinc, these products led to an increase of the content. In particular, in the case of Rhizosum PK, the improvement of these nutrients was statistically different compared to the control. No significant variations were found for the productivity parameters, for the yield, and for the grain quality parameters, but the most important results were found in the gluten protein subunits. In particular, the 3 products led to a significant improvement of the 81 kDa of the high molecular weight glutenins and to an improvement of the 43.6 kDa of the low molecular weight glutenins. These results are important because these gluten protein subunits improve the technological quality of flour. So we can say that the biofertilizer the biofertilizers led to an enhancement of plant growth and nitrogen nutrition. They allow it to alleviate the nutrients deficiency, in particular phosphorus and iron, that are very useful, can be very useful in the alkaline soils. And despite no improvement in the grain yield, the most important results were found in the gluten quality due to upregulation of specific gluten protein subunits. Another way to improve the sustainability, reducing the chemical nitrogen amounts, is the foliar application. Compared to the soil application, the leaf application of nitrogen to the plants allowed to have a lot of benefits. As a higher efficiency, it has a faster uptake and nutrient translocation. It is less dependent by soil conditions, in particular in drought conditions, and it allows to mix the fertilizers with other agrochemicals in order to reduce the costs. For this reason, several open file experiments were carried out in order to evaluate the yield and grain quality parameters in wheat, comparing the conventional nitrogen management with the soil granular application with different doses of nitrogen amount applied through the foliar spraying during the cycle of wheat. In a first 2 years experiment in open field, we have compared the conventional management with a total nitrogen rate of 160 kilograms of nitrogen per hectare with 3 leaf theses, differing in the total nitrogen rate 96, 80, and 64, and differing by the nitrogen rate applied during the cycle 16, 12, and 8. For the soil application, it was used granular ammonium nitrate applied by hand, while for the foliar application, it was used liquid urea ammonium nitrate applied through a sprayer. No significant variations were found in the productivity parameters, in particular for the harvest index and for the 1000 seed weight in both the years, and also for the yield in the first year, while in the second, the medium and the lowest rate were statistically reduced compared to the control. A similar trend was also found for the grain quality, in particular for the Zeleny index in both the years, while in the wet gluten, all in the first year, all the leaf theses were statistically reduced, while in the second, no variations were found. The protein content was practically the same in the first year, while in the second, only the medium rate was statistically reduced. Anyway, the most important results were found in the gluten quality. In particular, the highest nitrogen rate, 96, was statistically improved compared to the control and compared to the lowest rate. It was found for the high molecular weight glutenins in both the years, for the ratio between the high and the low molecular weight glutenins, for the total glutenins, and for the ratio between glutenins and gliadins. As regards the nitrogen use efficiency, the best improvement and the best values was found by the lowest rate. This was found also for in both the years and also for the nitrogen uptake efficiency. On the contrary, the control has reached the worst value in all the cases. We can say that nitrogen use efficiency and the nitrogen uptake efficiency improvements are related to a decrease of the nitrogen rate. We can say that the marked reduction of nitrogen fertilization through the foliar spraying is feasible without compromising the yield and grain quality. The reduced nitrogen doses, minus 40% as maximum, can improve the bread making quality on floor. And despite the greatest improvement of nitrogen use efficiency, the lowest rate, 64, is not enough to have good performances in the productivity and baking quality. For this reason, in order to find the best performing treatments, another experiment was carried out. This time, we have compared the conventional management with the usual total nitrogen rate and an absolute control, and three leaf theses, differing in their total nitrogen rate, 96, 104, and 120, and mainly differing by the nitrogen rate applied between botting and heading. That are the most important stages for increase the protein content. Additionally, only in the first year, two different fertilizers were compared, the liquid urea ammonium nitrate and the granular urea that was dissolved in water at ambient temperature and then sprayed to the crop. Going through the results, with the UAN, the medium rate has allowed to increase the productivity parameters, in particular a statistical increase for the yield and for the hectoliter weight compared to the control, while with urea, it was the lowest rate, 96, that has reached the best increase for the yield and for the harvest index and for the 1000 seed weight and also for the hectoliter weight. On the contrary, as regards the grain quality, for the protein content, no variation by a statistical point of view were found for the UAN, while in the other cases, for the urea and for the other parameters, wet gluten and Zeleny index, were the medium and the highest nitrogen rates that has allowed to increase all the values and the parameters. We can state that the lowest treatment, the 96 kilograms of nitrogen per hectare, has determined appreciable increase only in the productivity results, while the medium treatment, 104, has improved only the grain quality without greatly improving the yield. Between the two different fertilizers, urea seems to achieve better results and it is recommended as being cheaper than UAN. After have discussed about the benefits of the foliar fertilization management, by an environmental point of view, very little information is available about the amount of nitrate leaching obtained by these two different practices. For this reason, a lysimeter study was set up during the last crop season, 2019-2020, aiming to estimate the nitrate leaching of 4 treatments. In particular, we have compared two different managements, the conventional management and the foliar application with a total nitrogen rate of 96, and two different soil types, silty loam and organic. The nitrate leaching measurement was assessed collecting the percolation water after heavy precipitation or rainy period, measuring the water volume percolated, and then the water samples collected was analyzed through the spectrophotometer in order to determine the nitrate concentration. Just a quick look to the meteorological condition. As you can see in the last crop season, the rainfall was very modest. Only high peaks of the rainfall were found in December and in June, but as you can see, the total leaching volume of water was relatively low and quite the same among all the treatments. As regards the total nitrate amount leached, you can see that in the organic soil, no great variations were found between the two different managements, while in the silty loam, a small increase compared to the control was found by the foliar application. The highest nitrate leaching was found at tillering. You can see here the highest amounts, and then this trend was progressively reduced during the crop cycle. Additionally, another small experiment was set up in open field in order to estimate the amount of nitrogen leaf fertilizer reaching the canopy during the cycle. You can see that the minimum value of fertilizer reaching the canopy was found at tillering, 51, and then this value was progressively increased during the cycle, reaching the maximum values at budding, heading, and flowering. This trend was related to an increase of the main green vegetation indices, in particular to an increase of the leaf area index, the green line, and to an increase of the cool area index, the yellow line. We can say that a sustainable reduction in nitrogen amount through the foliar application was not immediately correlated to a reduction of leaching, of the nitrate leaching, and the different soil properties, but in particular the modest rainfall, can probably have underestimated and affected the leaching of nitrate.
Sorry, Manu, you have 4 minutes.
Okay, thank you. Further investigation should be useful for have better insights between two different managements. Continuing to discuss about the environmental topics, the fertilizer inefficiency in the environment is mainly due by the mismatch between the fertilizer addition and crop demand, and by the poor ability to sense the plant and the soil nitrogen content. For this reason, the precision agricultural industries try to find and to test smart soil nitrogen sensors in order to help farmers to decide when to fertilize. In particular, to add the fertilizer before the deficiency symptoms in plants start, and don't add the fertilizer when the soil has sufficient nitrogen available. For this reason, during my period abroad at Bangor University, I have followed a project regarding the development and the test of nitrate EO-selective electrodes that are a sensor for the determination of nitrate in the soil. The project regarded a first step in which we have to develop and test the sensor, and then to combine this sensor with the plant and canopy nitrogen sensors, along with geostatistical and mathematical models for elaborate a decision support tool. In my part, I have tested and evaluated the effectiveness of this prototype for the real-time nitrogen measurement in a maize pod trial under controlled conditions. The trial was set up in a temperature control room, and after the fertilization, two treatments were compared. The control, in which the plants have received no nitrate in the Hoagland nutritive solution, and treated, in which plants have received nitrate in the nutritive solutions. Going through the response of the sensor during the experiment, you can see that after the sensor installation and after a period of stabilization in the soil and after the addition of Hoagland solution, the sensor were able to detect the different concentration of nitrate in the soil. Here you can see that in the control, the trend remained almost the same, while in the treated plants, they were able to quickly and goodly detect the different concentration and the variation of nitrate concentration. During 10 days, the plants were not watered in order to understand the behavior of the sensor in drought conditions. And you can see that below 22% of the soil moisture, the sensors were not able to detect the real concentration of nitrogen. And this increase of the response was due to the difficulty in maintaining a good contact between the sensor membranes and the soil. Just a quick look to the spud content, you can see that after the fertilization, there is a real good difference and statistical difference between the treated and the control plants for the spud content. And you can see here in the different color of green, or the different color between the treated and the control plants. We can say that the sensor gave good performance and results also in the confined setup and under controlled temperature. This method can replace the laboratory sample analysis and can be a good method for monitoring nitrate in the soils, and the soil and the climate, in particular moisture, temperature, and rainfall, are the main factors that can affect the readings of the sensors. For this reason, other trials in real field conditions are necessary to validate the sensors. In conclusion of my work, we can state that the commercial biofertilizers can be effectively exploited in sustainable wheat cultivation, with positive benefits in nitrogen nutrition and floor quality. Significant improvements in grain quality were found by the foliar spraying, and a nitrogen reduction by 25 to 35% up to a maximum of 40% is feasible without greatly compromising the main agronomic parameters. And the prototype nitrogen sensor can be an innovative tool aiming to help farmers to decide the real-time and site-specific nitrogen application. For this reason, the strategies evaluated have expressed appreciable results and positive advantages to achieve the agronomic and sustainable enhancements in cereals cultivation. I have concluded. Thank you for the attention.
Okay, thank you for presentation, Manuel. If there are some questions, first I start with the professor's laptop.
I have actually several questions, and I don't want to take so much time, but it's a little in kind of my also area of interest and research. So thank you, Manuel, for your presentation. It was a comprehensive study, And I will start just with like you logically presented in the first series of experiments where you presented results for biofertilizers. Was it somewhat surprising that you got this increase in nitrogen content as well as in shoot dry weight during these early vegetative stages? You know, because nitrogen was that a surprising result for you, Manuel?
No, it's not surprising because
in the end there was no effect on yield, but was it expected like a result to get this increase in nitrogen content during vegetative stage?
Yes, we expected to increase the nitrogen content because they are biofertilizer that are self for improving the nitrogen content in the plants.
But you got also in those without this nitrogen fixing bacteria, yes?
Yes.
Also. And was it also that you got this improvement in bread making quality? Like because this high molecular gluten, was it also like anticipated result?
No, they have.
No, I was just like surprised because you get quite an improvement because Bologna is already like it's also the highest quality bread making quality also here in Croatia, not just in Italy. So it was I was like kindly surprised. Okay, I can move also that to the second one because I see that you were also looking for this increase in the bread making quality as well as better optimum fertilizer management. And the most interesting are your actually results from the second series of experiment where you compared foliar, you know, and granule application. Yeah. And it's really surprising because you decrease the total amount of nitrogen by almost 40%, and you got a comparable results relatively. So it's a lot. It's a large amount. Can you tell me about the soil where you conducted experiment? Could you consider that this is like relatively fertile soil with like a high fertility maybe?
Yeah, it's
Do you understand? Because it also affects this carryover effect, because then you can decrease a little bit also amount of nitrogen. What do you think? Is it also the soil was a little bit?
Yeah, we think that the soil of the farm are rich in nitrogen, and you can see also here for the absolute control in which you can see that the absolute control has obtained good results also.
Excellent. And you can just like on this table, can you explain me a little bit, because you are showing it, why the harvest index values are so relatively low, about 36 and like that, in 2018 and 19? These values, you know, are 35, 30, that's a kind of, I don't know, like why? Is there some reason, a specific reason for that?
Probably the meteorology conditions, because during this crop season, during April, May, and June, there were a lot of rainfall and temperature was not so high, so probably may have reduced the harvest index and probably also.
No, I have to notice, sorry, Manu, I have to notice because we usually, we can hardly get harvest index below 42, whatever happens, even during some sales. So these are also very interesting results. And I'm very happy with that. I'm also maybe just for consideration that maybe that soil, because you got a really effective fertilizer management with such a decrease in the total amount of nitrogen using, you know, just foliar application, UN. So that was very effective. And then on the third type of research where you also expressed, because it was a specific growing season, can you give me more explanation? What do you mean by organic soil? What is the difference between silty loam and organic?
Silty loam is the soil of the experimental farm. We can say that.
Okay. And what does it mean, organic?
Organic is rich in organic substance and like a.
Is it, so the main difference was in organic matter or something else?
Sorry?
the main difference was in the organic matter or maybe something else?
The main difference were fine more for the nitrate leaching. No.
Yeah, I was thinking about the soil, like when you mentioned because you're probably expecting more leaching on which type of soil? I will just rephrase my question.
Okay.
On which type of soil you expected more leaching?
In the organic soil, because probably there is the mineralization of soil nitrogen that can increase the nitrate leaching. And probably because organic soil has a higher increase of nitrogen compared to the silty loam, that probably can have enhanced the leaching of nitrate.
Okay, and in the end, I would like to support this idea of like censoring nitrogen, like nitrate content in the soil, because it's the most interesting. So some additional research. Thank you very much. It was very comprehensive. It was a serious experiment, a lot of data to be presented, and thank you very much, Emmanuel.
Thank you.
Congratulations for your work. Thank you.
Okay, so we have to move to the next. Please, Manuel, unshare your screen.
Yes.
Thank you very much again.
Thank you very much.
If there is no other question, Alessandro.
Oh, it's okay. We go ahead.
Okay, otherwise we can have additional
The next presentation is Rashita Govid. Topic is peptides derived from Trichoderma peptaboil as alternative method to control rice blast and fusarium heat blight of wheat.
So, am I audible to all of you?
Okay.
Hello?
You can...
Yeah. So, hi everyone. I am Rakshita Govind and I worked on the project entitled Peptides Derived from Trichoderma peptidol as Alternative to Control Rice Blast and Fusarium Head Blight of Wheat. I worked under the supervision of Professor Luca Sella. So let me start with brief introduction about my work. Actually, wheat and rice are two most important food crops which are used all around the world. Together, they contribute 50% of world cereal production. And also, as the world population is increasing very rapidly, it is very important to increase the production of these crops. But there are several reasons which are like causing the decrease in production of these crops, and the most important reason is the disease caused to them. So considering about disease, rice blast disease and fusarium head blight are such diseases caused by phytopathogen Pericularia oryzae and fusarium graminearum respectively. So the rice, let me tell you something about rice blast disease. It is very common in temperate rice growing regions. It is distributed all around the world. It causes 10 to 30% of yield loss per year globally. And if the cultivar is susceptible and environment is favorable for the fungus, it could lead even up to 100% of production loss. Therefore, Pericularia is considered scientifically and economically the most important fungal phytopathogen, and it can affect any part of the rice plant. As we can see from the picture, this is special like diamond-shaped lesion which is caused on rice leaves. Similarly, its fusarium head blight is also distributed all around the world. It causes yield loss, and the problem occurs when the seed grains are get contaminated with mycotoxin, especially the dawn. The dawn causes two types of problem. First, health problem to human and animals upon consumption, and another is technical problem, which is related to when these dawn contaminated grains are used for any enzymatic process such as beer production or something. This affects this dawn contaminated grain is problematic at that time. So to manage these diseases, there are several strategies available that I have mentioned here, but the most common strategy is the use of fungicides. But as we know, fungicides have another some disadvantages to the environment, and for that reason, they are not allowed in all the parts of world. So apart from them, biocontrol agents have found to be very effective, especially the Trichoderma species, which have shown high satisfactory degree of control of disease in lab condition. They use three types of mechanism, namely niche exclusion, mycoparasitism, and antibiosis to fight against the phytopathogen. But the application of these trichoderma spores in the field environment is limited because of several reasons, such as pH, moisture, temperature, and soil type. All these factors and other as well affect the viability of the spores in the field condition. So we decided instead of using spore into the field environment, we will exploit one of its mechanism, that is antibiosis, and we will use it for the protection of our crop. So considering this, we got to know about peptide balls. Peptide balls are the molecules that are naturally produced by trichoderma species as secondary metabolites. These are found to have antibiotic properties. These are short non-ribosomal linear peptides and rich in non-proteogenic amino acid. So with these things, we decided our objective as first trichogen GA4. It is a peptide ball. So it is naturally produced by Trichoderma longibrachiatum. So the chemistry group of our Padova University synthesized different analogs of trichogen. So we decided first to screen the analogs of trichogen GA4 against our pathogen of interest in vitro condition. And then those in vitro effective peptide balls, we decided to test their efficiency on model and host plants, that is wheat and rice plants. And actually we got nice results here. So we were interested to know how these peptide balls are affecting our fungus. So we did microscopic analysis, and finally RNA-seq analysis we also did to understand the molecular response of the fungus towards these treatment. So this is a trichogen GA4 sequence, which is produced by Trichoderma longibrachiatum. And based upon these template, different peptide analogs are synthesized by the chemistry group by doing some kind of modification, such as substituting lysine instead of glycine and so on. So based upon these objectives, I performed experiment where I did in vitro screening against, first let me tell you about Fusarium graminearum. I used only one strain, 8x1, for this screening. I basically inoculated peptide balls. I inoculate fungal conidia and added peptide balls in respective wells and compared the growth of mycelia with the control in which no peptide ball was added. And the result was quite interesting, that except for peptide 3 and peptide 19, all were very effective in reducing mycelium growth, even at 15 micromolar concentration of peptide. So we selected these peptides to be tested on plant directly. Similarly, I did with Pericularia oryzae also, but here I used 4 strains. First, Guy 11 from French Guiana, then AP and IT10 from Italy, and I203 from Vietnam. So here, here we have 4 strains to be tested against different peptide balls. So here I'm showing you a combined graph. This bar is for AP, then this light gray is for Guy 11, this dark gray indicates I203, and finally the black one is I10. So we observe different response of peptides against our different strains. For example, if we talk about peptide 3, here three strains are we found that 3 strains are inhibited, but not one strain. Similarly goes with peptide 6. 3 were reduced in mycelium growth, but not one strain. So based upon this observation, we characterized our peptides into inhibiting peptides, partially inhibiting peptides, and non-inhibiting peptides. So inhibiting peptides were those which allowed the mycelial growth of all the 4 fungus only up to 30%. And partially inhibiting were those which allowed the growth of all the 4 fungus up to from 30 to 50%. And those which allowed more than 50%, those were non-inhibiting for us. So these were our inhibiting, and these 2 were in partially inhibiting peptide balls, and we use all these peptide balls to be tested in plant conditions. So coming to the results efficiency, where I determined the efficiency on model and host plants. First, let me tell you about wheat. So I made a suspension of fungal conidia added with peptidol and sprayed on wheat spikes. And I observed that now the peptide balls which were effective in vitro condition, not all were very efficient in vivo as well. So peptide 5 was the best in controlling the bleaching symptom of wheat spikelet, followed by the peptide 4 ring. Here we can see in the picture, this is control where no peptide was added, and it's completely bleached after 21 days of inoculation, and peptide 5 remained green. It seems as if there is no symptom of bleach of Fusarium head blight. So based upon this, we also determined that if what is the DON concentration on our spikelets, and we observed that peptide 4 ring and 5 had very least amount of DON compared to control and other peptides such as peptide 7 and peptide 22 treated wheat spikelets. So these were having very less, which corroborates our finding here, peptide 5, which were best. Then we moved to pericularia. So for pericularia, I performed experiment for in vivo experiment first on barley plant, because barley are a model plant to study pericularia oryzae infection. I did here point inoculation, and in control I didn't add any peptide, and in treatment I added peptide. And here also I found difference in efficiency between in vitro and in vivo. So these blue color highlighted were the best peptide that shown nice inhibition. Here we can see from the picture as well. In case of peptide 4 ring, we didn't observe any kind of lesion, whereas in others we found something. Similarly here, peptide 2, no lesion was observed. So I measured the area of lesion, and based upon this I plotted that graph. Now these peptides were very effective, so I wanted to test if they are really effective on host plant also, and that is rice plant. So here I did the same experiment, the point inoculation, but instead of calculating the area of infection, we developed a disease index based upon the literature, and we have 5 level of disease index, and we found that all the peptides that were effective in vivo on barley were effective on rice also, but the severity of disease were more as compared to the barley. So this is my result for in vivo experiment. So all those which were effective on barley were found to be effective on rice also. After that, we did microscopic analysis to understand what physiological changes that this peptides bring on our fungus. So I did 2 type of microscopy, optical microscopy and fluorescent microscopy. So let me tell you first about optical microscopy. So this is the normal conidia that is without treatment with peptide. And here we found that the cytoplasm of conidia is evenly distributed all over the cell. But in case of treated peptide, we found that the cytoplasmic material got densely agglutinated at one place. So there was clear difference in the morphology of untreated and treated conidia. Also, we observed that untreated conidia were germinating after 48 hours of incubation, but treated conidia seems to be germinated but lies, as if all the cytoplasmic material have extruded out, something like that. So based upon this, the clear differentiation in morphology, we wanted to know what is the exact location of our peptides. What is the main target of our peptide? So we asked chemical group to attach fluorophore molecule with one of the best peptide, and then we will treat it with the fungus and try to see in fluorescent microscope. And this is the same we found in Fusarium also, that Fusarium developed mycelia, and the treated Fusarium found to have densely agglutination, same as the pericularia. So for fluorescent microscopy, this is a picture of conidia treated with Fitch conjugated peptide. So this is a optical microscopic picture. This is what we see under optical microscope. And when we observed in fluorescent microscope, we saw this kind of image, which seems that our peptide gets localized on the cell wall and also in the cytoplasmic condensed agglutinated cytoplasm also. Apart from this, we observed another interesting thing was that when we observed normal conidia in our normal conidia, like without treating with peptide, no Fitch molecule, we just observed in microscope. This is untreated conidia, and then we found as if they are growing normally, they're germinating. And when we observed this in a fluorescent microscope, as expected, we didn't find anything because there were no chlorophyll molecule, no Fitch molecule. But when we treated our our conidia with peptide, not conjugated with Fitch, we observed this in microscope, in optical microscope. But when we observed the same thing in fluorescent microscope, we observed some kind of fluorescence. So later we realized that this is autofluorescence, which indicates that cell is no more viable, the cell is dead, so it starts to flourish. So this is autofluorescence. And after that, finally, I did RNA-seq analysis to understand what goes on molecular level, what happens to the fungus. So basically, we treated mycelia, pericularia mycelia, with peptide 4 ring at 50 micromolar concentration for 3 and 6 hours. In control, we didn't make any treatment. And we found several genes to be up and down regulated, the differential expression of the genes. So this is the picture showing up and down regulations of the gene. We found most of the gene up and down regulated genes. And based upon this, we did some kind of characterization, like we understand some functions. We tried to understand what is going on how fungus is going to respond, and we hypothesized that it affects it's the peptide generates ROS, affects the upregulation of ROS forming genes, which causes H2O2 burst or any hydroxyl ion increase in hydroxyl ions, which causes oxidative stress, and that oxidative stress causes irreversible damage to membrane, mitochondria, protein, and lipid, and other nucleic acid. We also observed the upregulation of several glutathione synthesis and degradation gene, which play important role in detoxification of toxic compounds, and upregulation of other transporter genes, which also play a role in detoxification of xenobiotics. After that, we found some genes are also affecting the membrane, and DNA damage are also getting upregulated. So we found upregulation of lipid biosynthesis gene that directly or indirectly affect the membrane integrity. Many differential expression of genes were also involved in DNA repair, remodeling, and maintenance of these genes that led us to hypothesize that our peptide caused DNA damage that lead to the cell death. We also observed some genes related to autophagy and proteasome. So early upregulation of genes involved in autophagy that indicate that our peptide could cause protein damage and induce or program cell death. After that, we also observed upregulation of several genes related to cell wall biogenesis and remodeling. So chitin and glucan synthesis are important genes which are involved in remodeling of the cell wall. So those were also upregulated. We also found the upregulation of melanin biosynthesis gene. So melanin acts like a protective layer around the cell wall, which strengthens the cell wall and prevents the direct penetration of peptide. And this is also we observed also. This is the vial connecting having mycelia, where mycelia is germinated, and this is not treated with peptide, so it seems light brown in color. But after 48 hours, the same vial turns in. After 48 hours, the treated mycelia found to have brown color, which seems that this is melanin deposition. So this is because of the melanin deposition in peptide treated. And here in microscope, we also observe the same. Our untreated conidia were germinating very nicely, but in treatment, we found that there are kind of barrier which is not allowing fungus to germinate, and we see we hypothesize that we might say that this is the deposition of melanin, this brown in different color. And so at last, I would conclude by saying that we found several trichogen analogs that were effective in inhibiting mycelial growth of Fusarium and Pericularia at 15 and 50 micromolar of concentration in vitro. In vivo, we found on wheat, peptide 5 were the most effective, followed by peptide 4 ring. And on barley that I did against Pericularia oryzae, 7 peptides were very effective in controlling blast symptom. And on rice, all the 7 peptides were affecting in controlling rice blast symptom also. From microscopy analysis, we found that peptide cause morphological changes in spores, such as membrane shrinkage, dense agglutination of cytoplasm that lead to the inhibition of spore germination. Also, we hypothesize that peptide can pass through the membrane and locate intracellularly in the agglutinated cytoplasm. And with RNA-seq, we found that when P. oryzae treated with peptide 4 ring likely cause stress in the fungus and rapidly induce the autophagic cell death. The fungus reacts by remodeling its cell wall and activating other transporters and detoxification mechanism, and which could be a direct or indirect damage to plasma membrane and DNA. So at last, I'm very thankful to all the people of my lab who helped me in this beautiful journey of my PhD, and thanks for your attention.
You, Rakshita, you are perfectly in time. Sorry, I didn't tell you when you had your 5 minutes left, but you are in time. So
okay, thank you.
Okay, thank you for presentation. I can start with some questions. First of all, about the efficacy of peptides against Fusarium graminearum on wheat spikes, this was highly variable compared to the in vitro result.
Yeah.
So what explanation can you give on the efficacy of peptides against graminearum in vitro with respect to in vivo or spike?
Yeah, we can hypothesize that there are several reasons for this. First, in vitro, I am directly inoculating the fungus in PDB medium along with the peptide. So they have a nice area, like they are immersed in the PDB medium and they are in contact with the peptide. So so the peptide can have can attach or have higher surface area to react with the fungal cell. But in case of wheat, I made a spray infection first. So we assume that this might be because that there is no proper attachment of peptide and fungus first, and also there are some difference in microenvironment as well. Like there are many other protease degrading genes or
Sorry, but there are some particular amino acid charged that can affect these.
Yes, we observed that like peptide 5, these were with three positive charged amino acid, which showed very good inhibition, followed by peptide 4 ring, which had 2 positive charge amino acid. So we can think of this that these positive charge peptides are causing death or leading to death of our fungus. We can say this.
Okay. Between barley and rice, you have found that the efficacy of treatment with peptide against Pericularia oryzae occur at different extent.
Yes.
So how can you explain why the efficacy of peptides on barley is close to 100%, while on rice it reaches 70 or lower?
Here we assume it is because here, as I can show you in the picture, in on barley we have wider leaf surface. Sorry. Yeah, here we have wider leaf surface. So when I inoculate on barley, the the suspension clustered together and form a bead, and it stays over here. So this because of the hydrophobicity of this leaf also. But in case of
But
yeah,
the reason of this cluster of drops depends by what?
probably because of the hydrophobicity of barley leaves, because they can attach more here. And here if we see the rice leaf, these are slender, so low surface area.
Because
and
there is some difference in specific epicuticular content.
Epicuticular surface, yes, of course.
Okay. Another curiosity, why the choice to test peptide on 4 strain of Pericularia oryzan from different part of the world, no?
Yeah, actually,
and
yes.
only one for Fusarium graminearum.
Yeah, basically it is because due to COVID, we didn't have much time to test more Fusarium spore. But actually for Pericularia, this project we started and we got nice results from the beginning. So we were interested to know that are these peptides are effective against fungus all around the planet, like one strain I used from Vietnam, Asian strain also. So
just on RNA-seq analysis, no? For this transcriptomic analysis on fungus, which responses are about your study determined by direct toxic effect of peptides, and which one could be more a consequence of an indirect effect?
Okay, so we hypothesize that actually our peptide causes lot of ROS to generate, and these lost this ROS molecules like H2O2 and other hydroxyl molecules causes cell death. So we assume that this might be because of the death might be because of ROS, or we don't know if it is because nucleic acid being negatively charged get attached with peptide positively charged, and the degradation happens. So we do not know here which is the main reason for the death of our fungus. So I can say that this is the damages that is caused to the membrane, mitochondria, and lipid because of the ROS generation might be indirect, and direct could be because that peptide can act on nucleic acids being different in charge, both.
But about your opinion, what is the biochemical chemical characteristic of the best peptide on the base of the chemical feature? Because containing lysine and other hydrophobic amino acid. So what is your opinion how to build up the best peptide?
Yes, we observed that the peptides with being more positive charged have found to be more effective. Like peptide 5, those were effective on both on wheat and rice plants, also barley also. So I can say that being more positive charge can be good can be good peptide ball having more positive charge. I can hypothesize this.
There is an explanation why at the 6-hour transcriptomic analysis identify few differential genes expressed compared to 3 hours.
Yeah. Okay. Actually, it's not that we see that we have a lot of gene expressed upregulated compared to downregulation. But this is probably because of, if I show you another one heat map, yeah. So here we can see that this is 3-hour treatment, where we have this is control and this is treated fungus, and we see nice cluster of like we have nice replicates of treatment and control, which gives us nice result. But if we talk about other 6-hour 6-hour RNA extracted, we found that we have difference in the replicates, you know, which might gives us false result. We are not very sure, but this might gives us false result because statistically it is not supported. So it's not like at 6 hours less genes are expressed. I mean, down regulated. It's not like that. It's because of the statistic we are getting this kind of result.
Okay, just the last one that is a curiosity in the application. To test peptide, you have co-inoculated them with the fungus. That is a condition that doesn't occur in field. Infection occur before or after treatment. So, for a practical point of view, use and timely application in field, what aspect do you think should be taken into account?
Yeah, first we have to determine this efficiency, like in our lab condition there. Like, of course, it's true that I made a suspension together with peptide and fungus, and I inoculated. So sorry.
So could be necessary to improve shelf life of this peptide?
Yes, of course.
You have an idea how?
Right now, know how to increase the shelf life, but yes, it is important aspect to be considered before going into the field environment, before testing the field environment.
Okay,
sure.
For me it's okay. It's to know if there are other questions.
No question for me, Zlatko, if you want to ask something.
Okay.
We still have few minutes if we want. There is the break now, so if you want, Zlatko, please.
Just a short comment. It is a general comment on all the presentation, because it is difficult to follow just jumping from the introduction into the almost results and the other sections of the presentation. It was just I would like to ask Rashida, like for the follow-up research, would she more focus on the research on the wheat or on the rice? Just want to hear her opinion.
In my opinion.
Yeah. Do you understand the question?
Yeah, which is you.
Yeah, if you want to continue your research, like after this like preliminary, which crop would you pick up for the follow-up research?
Actually, both crops are important, but as I read from the literature, and Fusarium is more, I feel it is my feeling that more dangerous because it leads to a lot of like 100% loss in crop production. So it is important to focus on rice crops and also not like
I was thinking that you are focusing more on the potential effectiveness of these peptides, you know?
Yeah.
Like what do you feel like for the future? Like where is more potential, on rice or on wheat? I just want to hear your opinion.
You mean to us that my peptide balls are more effective on rice or sorry, against the pathogen Pericularia or Fusarium?
Are there any potential in the future?
Yeah, it has more. Sorry, can you please repeat question once more? Actually, I'm getting a little confused.
Yeah, so you like from your introduction section, you pick up these two crops mainly because these are two staple crops in the world, yes?
Yeah.
That was the main how to rational beyond that. So you got your results from this research, and if you have the opportunity to continue your research, would you focus on the effectiveness of these peptides on wheat or on rice? Where is more like potential for any, how to say, for any practical application?
From practical application point of view, I got nice result with wheat, to be honest. Like peptide 5 was very much effective.
That's enough. Thank you very much, Rakshita. Thank you very much.
Thank you.
So, okay, Rakshita, please unshare your screen. Thank you very much.
Thank you.
And we would like to make a short break, 10 minutes, let's say. We'll start again with the next PhD student, who is Matteo Longo. So Matteo, be ready. And at 20 past 11, okay?
Yeah.
Rakshita, stop your screen sharing, please.
I think she left.
Yeah, she disappeared, but she have to.
Sorry, Prof. I'm here. Actually, I have the problem with the.
There should be a red.
Yeah, but I'm not able to join it. I don't know why. So just a moment. Yeah.
Okay, thank you.
Sorry, is it now really a coffee break for all?
Yes.
Yeah,
I believe so. Yeah. So I could. Just also. We are also supposed to leave for a coffee. Okay.
Okay, 10 minutes.
Okay.
Eccoci qua.
We are back. Okay, Matteo is ready.
Hello.
And so we can, Zlatko is back. Okay, we can.
Trying to share.
Sorry. Matteo,
Wait.
you can start.
Okay. Okay. Hello, everyone. I am Matteo Longo, and my supervisor is Professor Francesco Morari, while my co-supervisor is Professor Nicola Dal Ferro. The title of my PhD thesis is Enhancing Experimental in Silico Methods to Evaluate Ecosystem Service Trade-offs in Agri-Environmental Measures. Before starting with the main presentation, let me give you a brief overview about the European Union policies about agriculture. Indeed, the European Union has many policies to address and to reduce the pressure on the agriculture and on the environment, and among them we can find the agri-environmental measures. That, in particular, for the Veneto region, our region here, the total rural development program has dedicated 20% of its budget for the agri-environmental measures that are here financed with the aim of increasing water quality, decreasing soil degradation, and greenhouse gases emissions. But how have been they sustained? Since their establishment, the agri-environmental measures have been sustained following an action-oriented approach, meaning that farmers are not paid for the provision of environmental outcomes, but for the adoption of land management practices. And this approach has been largely questioned, among other things, to poorly targeted environmental issues and a lack of site-specific payments. However, considering the high area and the high budget involved, as an alternative, a result-oriented approach has been suggested in order to better allocate the payments and maximizing the ecological benefits. And following this approach, farmers will be paid according to a scientifically based evaluation that should lower the mismatches between the payments and the environmental targets. However, developing such a scientifically based evaluation using only direct measurements, so empirical and local studies, will be almost impossible for regions or countries. So as an alternative, as a solution, we could use biogeochemical models. These models are process-based, means that they mathematically represent physico-chemical and biological processes, and therefore they differ from empirical models that they just rely on experimental data. And among the advantages of these models, they can easily overtake special limitations of local empirical studies and the expense of the direct measurement. Indeed, they can be easily upscaled over wide areas such as regions, countries, continents, also the entire world. And in addition, they can combine different biogeochemical cycles, so nitrogen, sulfur, carbon, and so on, and assess the fluxes to and from the components of the environment, so air, soil, and water at the same time. On the other hand, they require careful calibration with measured data and a critical validation in order to assess their robustness. Where for calibration, we mean the adjustment of the influential model parameters in order to obtain realistic results. And for validation, we mean the comparison of the outputs from the model with the additional observed data that are therefore different from the subsets using for the calibration. Considering that the open questions, the gap we aim to fill concern the accuracy of the current models to reproduce the peculiar conditions of the Veneto region. If the in silico methods, therefore, models can contribute to offer, can contribute for evaluating the advantages and disadvantages of the measures. And finally, if models can be useful to drive a transition towards a result-based approach. Therefore, the specific aims were to implement models on the peculiar pedoclimatic condition of the low-lying Venetian plain, calibrate models with a holistic approach that considers all the main biogeochemical fluxes, and finally quantify at fine spatial scale the trade-offs among some ecosystem services of the measure implemented in the Veneto region. The thesis was developed in 4 chapters, but now I'm going to focus just on 3 of them. The first one was about the model implementation under peculiar conditions. Among the most concerning problems related to biogeochemical models, the cascade model and the water description is one of them. Specifically, most of models use the cascade model to describe the water dynamics. And using this model relies on the concept that each soil layer has a specific drainage storage, and when the water exceeds this storage, that is the field capacity, percolation and drainage starts. This is an easy and fast method, but it does not allow to simulate saturation and also upward fluxes, because the water just moves downward. This is especially problematic under shallow water table conditions, where saturation and capillary rise effect often take place. And this is something that often occur in the low-lying Venetian plain. So using cascade models, it is very difficult to predict with accuracy the soil water content. To overtake this problem, we could use hydraulic models that were specifically designed to solve the Richards equation. It is the main law that describes the movement of water in saturated and unsaturated soils. However, this is a highly nonlinear partial differential equation, and therefore it is computationally very expensive. The aim of this chapter was to implement a fast solution of Richards equation coupled with a biogeochemical model. In order to do that, we needed experimental data, as we saw from the second slide, and we retrieved the data from the experimental station of the University of Padua, where a set of lysimeters were set up and highly instrumented to govern the boundary conditions and to monitor the soil water dynamics. The data collected, the data used were from 2011 to 2020, and across this period, many factors have been tested in that lysimeter stations, but the water table level was kept fixed across all the period, and the treatments involved were free drainage conditions, water table level at 60 centimeter depth and at 120 centimeter depth. In total, we used data from 12 lysimeters. As a model, we chose EPIC. This model was developed in 1984 with the aim of assessing the impact of erosion on crop productivity. However, it's been continuously implemented and expanded, and now it is able to simulate several processes on a daily basis, such as nitrogen leaching, pesticide fades, or organic carbon dynamics, and so on. Recently, it was implemented with a fast solution of the Richards equation based on the Ross method and a microbial denitrification on an hourly basis. The setup we used for the soil water dynamics involved 2 different methods. The first one, as I already said, was a Richards equation solver using the Moelle and Van Genuchten model, and the second one was the variable saturation hydraulic conductivity method. That is an upgrade of the cascade model approach, where it simply reduces the vertical hydraulic conductivity based on the saturation and therefore the soil water content. The Richards equation is usually numerically solved through several iterations of nonlinear equations. However, the Ross method implemented into EPIC yields sets of linear equations that can be solved without any iteration. And this allowed the model to be up to 45 times faster than iterative methods. The experimental data that we used for the simulations were split into calibration and validation subsets. Specifically, the soil water content from 2013 to 2017 were used for calibration, while the last 3 years for validation. In addition, we also used annual fluxes of percolation, evapotranspiration, and upward fluxes to evaluate the performance of the models. Here are some results about the soil water content simulated. Here I'm showing in this graph, you can see the soil water content observed simulated with the 2 methods arranged by the groundwater conditions, so free drainage and the 2 shallow water tables, and by the depth of the soil probes. As we can see, the Richards solver so the dark green, well performed for under all groundwater conditions, even under shallow water table. On the other hand, the cascade model well predicted soil water content just under free drainage, while under shallow water table levels, the soil water content were highly under predicted. That is the light green line. However, both methods sometimes misrepresented soil water content, especially related to the yield that was simulated by the model. For example, here we can see an under prediction due to a yield over representation. The observed one is the purple line, and another prediction of the soil water content due to yield over representation, which in turn increased the evapotranspiration. Evapotranspiration is one of the fluxes that is most important to evaluate for these kind of models. And here we can see that the simulations of the Richards method were much better than the cascade models, in particular for shallow water table conditions. While for the percolation, both methods over-predicted values under specific conditions, as we can see from the average simulations and for both methods. In particular, free drainage over-predicted, in particular the cascade method over-predicted percolation under free drainage. Finally, the upward fluxes from the water table were misrepresented by both approaches, in particular the WT60. To conclude this part, soil water content and evapotranspiration for free drainage were similar using the two methods, and this suggests that using the cascade model could be better for simplicity and efficiency under such conditions. Under specific conditions, percolation failed for both approaches. And finally, the Richards outperformed lysimeters under shallow water table, both in terms of soil water dynamics and evapotranspiration. EPIC is now able to simulate the peculiar hydrologic conditions of Veneto region after the Richards implementation, despite some little issues still persist. The second chapter was about the comprehensive simulation of nitrogen and carbon cycles. Specifically, after the very good performance that we got in the water description, we also wondered if EPIC was also able to simulate such fluxes with the same accuracy. To test the models, we used experimental data retrieved from 44 fields, and specifically from 3 farms spread across the region, so different pedoclimatic conditions, and involving 3 management systems: conventional agriculture, cover cropping usage, and conservation agriculture. Therefore, different processes involved, where the last 2 of them are also agri-environmental measures financed by the Veneto region. Across the experiment that lasted 7 years, and therefore also the simulation, many kind of data were collected, but now I'm going to focus on the soil organic carbon stocks on 2 different years and on the crop yield. Before starting the simulation, a screening test, the Morris method, was performed in order to identify the most sensible parameters, and such parameters were then calibrated using an automated algorithm. 2 different nitrogen and carbon submodels were tested and compared inside the model. The first one is a century-based model that is based on the concept of pools, and it is considered among the state-of-the-art for SOC modeling, but it does not involve the microbial biomass to drive the decomposition. The second one is a Phoenix-based model that is still based on pools, but it does involve the microbial biomass on some processes. Here are some results about the yields. All the management systems were well simulated by the model, especially the conventional agriculture, as we can see from the R-squared. However, despite the high variability in the observed data, that could be partially explained by some missing information about field and crop weed competition. All of that considering that we also added the presence of sorghum alepenses as weed in conservation agriculture in order to account for important weed infestation episodes that were experimentally observed. And as we can see from this graph, the the performances of the model greatly improved about the addition of the yield. About some results about the soil organic carbon stocks, both submodels, so the century-based and the Phoenix-based model, very well predicted the soil organic carbon stocks, both considering them by layers and in the whole profile, so from 0 to 50 centimeters, and also considering all the different management systems. The behaviors among them were very similar. To conclude this part, EPIC offered good crop simulations considering the high variability among different fields. The with addition to the simulation model improved the conservation agriculture yield performance, and the model showed its ability to perform well across systems and years by capturing the soil organic carbon stock variability affected by the tillage systems over time, and by highlighting its potential for SOC-related ecosystem service evaluation.
Matteo, just 5 minutes, more or less. Okay.
Thank you. The last chapter on which I will be focusing is about the site-specific quantification of the A&M's trade-offs. And specifically, we tried to answer to the last open questions we had. And we did that through a site-specific modeling, in which we coupled biogeochemical model with georeferenced territorial databases at high resolution, such as climate, crop management, fertilization, irrigations, and so on. And in total, we ran 84,000 simulations. To evaluate the performance of the measure, we compare agronomic and environmental outcomes of scenarios with and without the adoption of the measures. And we compare the nitrogen use efficiency, and we also developed an integrated index that included the combined effect in terms of yields, soil, water, and air quality. The nitrogen use efficiency was calculated in following the approach suggested by the European Union Nitrogen Expert Panel, in which it's the ratio between the nitrogen removed by crop and the total input of nitrogen. And following the suggestions of the expert panel, we identified a desirable range that was between the value of 0.5 and 0.9 nitrogen use efficiency, and we also excluded the nitrogen surplus greater than 80 kilograms. Here, in the business as usual scenario, so without the simulation of agro-environmental measures, 76% of values was inside the desirable range, so a good result. Here, I'm showing you the nitrogen use efficiency for some measures, specifically conservation agriculture, organic farming, and meadows. Here, we can see that the limited nitrogen input and the tight crop rotations lowered the nitrogen use efficiency, both for conservation agriculture, as you can see here, and also for organic farming. And 94% of the agro-environmental measures produced news within the desired range. About the index that we developed here, you can see some maps about those performances. Specifically, the blue polygons shows the measures able to deliver ecosystem services, while the red or orange polygons, there was an undermining of the ecosystem services. Using this method, our approach was able to recognize the effect of the measures in a spatial context, and ecosystem services, as you can see, were substantially enhanced, as you can see from the blue color, the predominant blue color, and organic farming had both increased and decreased values, so both colors, while the best strategies included permanent soil cover and minimum soil disturbance. So conservation agriculture and pasture and meadows. To conclude also this part, the model implementation at the fine spatial scale was able to assess the performances of the measures and to disentangle between pros and cons from the adoption. A positive picture generally emerged on the effectiveness of the measures to deliver essential ecosystem services in the Veneto region. And finally, the effectiveness of some measures was different in a spatial perspective, highlighting the need to target local strategies. The overall conclusion, when accurately calibrated biogeochemical models such as EPIC adapted even to the peculiar conditions of Veneto region, a comprehensive calibration and validation against experimental data is essential to model with accuracy the water dynamics and to reproduce the main carbon and nitrogen fluxes. And this site-specific modeling approach proved to be a suitable tool to evaluate the implementations of the measures using direct assessment of the ecosystem services. Finally, the models demonstrate to be optimal tools for a future development of agri-environmental measures using a result-based approach. And with that, thank you for your attention.
Thank you, Matteo. I just have a question, if I can, Alessandro.
Okay.
Can
I don't see you, sorry.
So anyway, I have a question, Matteo. It's a general question, okay? Based upon your final consideration, how do you see the next step? I mean, you say that your modeling approach was effective, and maybe you have to set up a practical tool to be able to put this model into the real context, okay? To be able to go really into a farm and say, yeah, okay, this is your final result. So this is the money that you can gain this year, okay? Which is the next step to arrive to that final?
Yeah, okay, that is exactly what should be the aim, okay, of this this work. Obviously, okay, this involves also the policy makers, so regions and so on. And however, what we would like to do, okay, is the developing such instruments, okay, improve them, and then we could also link that, okay, with the yield and the money, okay, that we can get, that the farmers can get, and therefore they can adjust the benefits and so on, how much money they should take. In this way, okay, we should be able to allocate more money where we really deserve, okay, and less money when
no, that's clear. That's clear. So as far as I understood, so you see,
okay,
you see that the main obstacle as a policy maker obstacle. Okay, so that's the main restriction.
Okay, I wouldn't say that is an obstacle.
I mean, sorry, Matteo, but practically speaking, technically speaking, maybe it can be realized by now, let's say.
Technically speaking, yes.
Okay.
Because
so
all the tools
you are technically ready, but it depends on the
because
on the
also because they already did that sometimes and somewhere in the European Union.
Ah, okay.
Result-based approach has been suggested, but now they've been like on some countries, some states in Germany or regions. I don't know exactly the
land.
Okay, and also in Slovenia. So
okay, so at the level of a European Union, it's allowed.
Yes,
and it's regulated. So it's a question of Italy, let's say. That's
of all the
okay.
policy makers. Yes.
we know the situation here. Thank you very much. Very interesting dissertation. Very interesting.
You're welcome. Thank you.
Maybe there is a time for a short question or comment.
Yeah, there is time.
Thank you, Matteo, for your presentation. I just wonder for the, I'm just interested because I had an opportunity to visit Veneto region quite a few times and like that, very happy. Did you, when you analyze your data, and I saw that you pointed out that
sorry, you muted your microphone.
I'm sorry. Well, I will start with. I visited Veneto region quite, and in your presentation, you pointed out that for Veneto region still there is a little bit surplus of nitrogen, depending on whether it's conservation or other agriculture, did you maybe identify some agricultural practices or something which are the most detrimental from the environmental point of view? Because you pointed out the best will be approach, I don't know, permanent pasture, but that's not possible, you know. But did you maybe identified some crops or maybe some fertilization management or like that, which you consider particularly, how to say, not beneficial from the environmental point of view? So thank you. In some environments specifically, because in the end you get into site-specific management, you know. Just a short one, please.
Exactly. And okay, maybe we can better see that here in the business as usual scenario. Yes, we did recognize that actually it was very linked to the, it was very site specific. Okay, so it was not just a concern of crops, also because all these values, okay, are the mean, the average of the nitrogen use efficiency for a single polygon. Okay, so there are many crops averaged there, and it was very connected to the soil conditions and also, of course, to the fertilizations. For example, there was the nitrogen leaching was greater under where obviously where the hydraulic conductivity was greater, so sandy soils and so on. There was also obviously it was dependent on the on some crops and some methodologies. Like maybe I can maybe yes, okay.
It was very demanding to analyze such a data and to make it's very difficult to make comprehensive, you know, like because this is a like multivariate analyze of so much data. So thank you very much.
Thank you.
Any other question? Alessandro, we're okay. We can move to the next one.
Okay. Yes, we can go.
Thank you, Matteo. Thank you very much.
You're welcome.
So unshare your screen, please.
Yes.
And we can move to Nebojša Nikolić. Are you there? Yeah, you're muted. Okay,
I'm here.
Okay, good morning.
Okay. Can you see the presentation?
Yeah, of course.
Can you hear me?
I will let you know when 5 minutes are left, okay?
Okay. Well, good morning, everyone. My name is Nebojša Nikolić. I'm a PhD student of the crop cycle. My supervisor is Professor Roberta Mazin, and the title of my PhD thesis is Precision Weed Control in Arable Crops. First of all, history of agriculture is the history of fighting the unwanted plants that have always accompanied the crops. We can find references to weeds in some ancient texts and holy scriptures. Some recent findings from Israel illustrate that some of the species that are weeds today in the same site were present there also some 23,000 years ago. What is important about weeds is that they are very good competitors that often have upper hand in fighting against the crops, and that they usually appear in patches across the fields. Although the interest in weed control appeared very lately, some rudimentary tools for weed control are thousands of years old. Quite a revolution occurred in the early 20th century with the appearance of the first herbicides that have since become the major tool for weed control. And today we might be at the dawn of a new revolution that would have modern technology as the new type of weed control. Today, weed control is mainly divided into two parts, the mechanical and the chemical one. The task of both is, of course, to destroy weed, yet they have different approaches for doing so. The mechanical control usually destroys physically the weeds, while the chemical one relies on the use of different compounds that can cause inner or outer reaction that will bring to plants' demise. Of course, they are not perfect, therefore they have some negative effects that for the mechanical weed control are mostly related to soil disturbance that can lead to a desertification in some cases, while the chemical control can cause environmental damage and health issues. They also differ in efficiency, where the chemical weed control is more efficient. It is also mostly used, and in some agricultural practices, such as conservation agriculture, it may be the only possible solution. Exactly because of this advantage given to the chemical weed control, the main focus of this work are the problems caused by it that mostly revolves around the overuse of herbicides and the so-called blanket spraying, which is the term used to define the spraying of the entire area regardless of the presence or absence of weeds. Therefore, the task was finding a solution within better herbicide use or integrated measures, therefore within precision agriculture. Precision agricultural approach for weed control in this work for both conventional and conservation agriculture relies on two different parts. First one is the site-specific part addressed through weed mapping using the unmanned aerial vehicles, and the time-specific part used using the emergence predictive model AlertInf for obtaining the cumulative emergence of weeds in time. Both parts combined allow us to create prescription maps containing the information for precision weed control. Or better said, they enable one of the definitions of precision weed control given by Gebbers and Adamchuk, which is to apply the right treatment at the right place and at the right time. In order to address the aforementioned issues and reach the established aims, several experiments have been conducted, which will be shown divided into individual chapters. Here, a commercial unmanned aerial vehicle, Tanafi, coupled with its default RGB sensors, was used to examine its usefulness for weed mapping. A survey was conducted on 24th of May 2019 at a height of 35 meters, providing an orthophoto with a good spatial resolution. Once the orthophoto was obtained, an open source program, SAGA-GIS, was used for weed detection. More precisely, the three algorithms incorporated in this program, which were the maximum likelihood classifier, artificial neural network, and the object-based image analysis. In order to create prescription maps, it was decided to divide the fields into cells of different sizes based on the technical possibilities for precision spraying. These cells were 0.25, 2, and 3 meter squares. Finally, for the creation of prescription maps, ArcGIS Pro Model Builder was used, following the scheme you can see here. I will highlight only the most important parts that are the necessary inputs, the orthophoto of the field and the classification made by one of the algorithms, the field division with one of the cell sizes selected, and the decision algorithm, that is the decision threshold, at what percentage of infestation should a certain cell be sprayed. Here we used three decision thresholds: more than 1%, more than 5%, and more than 10% of infestation. Finally, an output is a prescription map which indicated only the cells which should effectively be sprayed. Here you can see some examples of those prescription maps. These particular ones are the result of the artificial neural network classification with more than 5% infestation. And you can see how the area to be treated changes with the different cell sizes. Here are some results about the possible spray area reduction. And as you can see, the classification changes within a single algorithm with different cell sizes with the same percentage. Of course, with the different algorithms and with the different cell sizes, but with the same percentage, the area also changes, and the area also changes with the same cell sizes, but with different percentages and with different algorithms. It is also important to notice the precision of the weed detection. Where more precise is the weed detection, there will be more area to be treated. Here we could see that the artificial neural network was the most precise method. Therefore, there is always more area to be treated using this algorithm. Still, the most important conclusion is that by using any of the proposed scenarios, the reduction of the area to be sprayed is very important. After realizing that it is possible to obtain good results with unmanned aerial vehicles and detection algorithms for weed mapping, we decided to add a temporal component to fully understand the potential of precision weed control and to assess the possible herbicide reduction. For this purpose, another survey was conducted over a maize field in its early growth stage in June 2019. Performing ground surveys before the flight, it was determined that the vast majority of weeds was in fact Sorghum halepense. This particular species is incorporated into the AlertInf program, and we were able to assess its cumulative emergence. And we have decided to conduct a flight in a date where AlertInf indicated 96% of cumulative emergence. The flight was conducted using an unmanned aerial vehicle DJI Matrice 100 coupled with DJI X3 and DJI X5 visible sensors. The same procedure for prescription maps creation was applied here as explained in the previous chapter, with the difference that here we use the artificial neural networks that showed the best results in the previous chapter, and the visible atmospherically resistance index VARI, which is one of the indexes integrated into the ArcGIS Pro program and whose usefulness for weed detection we want to test. Results show that the spray area reduction can go from 65.29% to 93.35% for VARI, and from 42.43% to 87.82% for the artificial neural network classification. Also here, by using any of the scenarios proposed, the reduction of area to be treated is remarkable. Herbicide use reduction is proportional to the reduction of the area to be treated, and to simulate the exact quantity and cost reduction, two herbicides were tested, Equip and Titus Mais Extra. We can see that the quantity to be applied changes with different classification methods, but is still much lower than the one that would be used with the blanket spraying. Also, the savings can be quite high, which is very important for the farmers, that is the final producers. I also need to highlight regarding my claim to the Dr. Europeus title that this work has been conducted in collaboration with the University Unilassal from Beauvais, France. Afterwards, considering the good results obtained for conventional agriculture, it was decided to test the possibilities of doing the same for conservation agriculture. Why? Well, conservation agriculture is a way of field management that is on a rise on a global scale. The map shows some of the main land areas where this type of agriculture is taking root, yet the latest data says that more than 180 million acres of arable land in the world is under conservation management, of which around 2 million are in Europe and 283,000 only in Italy. Conservation agriculture is different from conventional agriculture. It reduces the soil disturbance to minimum. It requires cover crops and crop rotations, and it cites a permanent residue cover of the soil. Therefore, weed control cannot be the same as in the conventional agriculture, and these differences need to be studied to be able to apply our methods also in this conservation, in this agricultural management type. To better understand these differences, considering how weeds are exposed to different conditions, an experiment to assess the changes in weed patches distribution between the two was conducted. For this experiment, two fields were chosen, one under conservation agriculture and one under conventional agriculture. An UAV survey was conducted once a year for 2 consecutive years in order to obtain the orthophotos. These were then classified using the maximum likelihood classifier for weed detection as described before. Once the weeds were classified, it was possible to confront the classification from different years using a similarity assessment, which is part of the raster calculator tool of the ArcGIS Pro program, which allowed us to assess the differences not just between the years in the same field, but also how does the weed distribution changes between conventional and conservation agriculture. What is interesting, and is of course a consequence of field management, is the area that remained equal in both years in both fields. The data show that in conservation agriculture, the area remained equal was 56.18% against 11.50% in conventional agriculture. And this difference is quite high and is the best illustration of the differences that exist between the two agricultural management. Another interesting point was the field margin that seemingly was unchanged in these years and was also moving inwards the field. Therefore, in order not to interfere with our calculations, we have decided to eliminate this part and to redo the calculations without it. Result difference also not even though not as high as in the previous example, is still almost the double between the two fields. With this experiment, we have gained a confirmation of site-specific differences, but these are also likely linked to time-specific differences that must also be addressed. Further on, we have decided to investigate the spread of weeds in conservation agriculture by testing the behavior of the seed bank in the no-till field, which was part of the conservation agriculture. The seed bank is the main source of infestation in conventional agriculture. Therefore, we were asking ourselves whether it has the same importance in the conservation agriculture. Considering the differences in management between the conventional and conservation agriculture and the presumable larger microbial activity in the conservation agriculture, we have decided also to assess this component and eventually to confront the results with an undisturbed habitat from whom we knew that there were much more microbial activity. For this experiment, 6 common weed species were chosen. For each, 16 steel mesh nets were filled with 50 seeds and buried at a depth of 12 centimeters, both in the no-till field and in the buffer strip. Exhumations were conducted after 3, 9, 15, and 21 months, and for each exhumation, 4 nets per species were taken out for germination tests. To measure the microbial activity, fertimeter technique was used, where the activity of the microorganisms was measured by the level of degradation of cotton threads for cellulolytic and silk threads for proteolytic microorganisms using a dynamometer. Fertimeters were placed a week before every exhumation to monitor the microbial activity, and in these pictures you can see how that actually looked like. The most interesting and important results are the differences between the degradation and the viability of the seeds between the two sites. As you can see, both the degradation and the loss of viability of the seeds for all species was always higher in the no-till field than in the riparian buffer strip. Some species, like for example, Alopecorus myosuroides and Digitaria sanguinalis, have completely lost their viability after 21 months and were also the ones that were mostly degraded. This is caused by the greater microbial activity in the no-till field, especially for cellulolytic microorganisms that were attacking the seed coat, exposing the embryo to both biotic and abiotic negative factors. We can also see that some species with thicker seed coat, like for example, Abutilon theophrasti and Sorghum halepense, were less degraded and had more viability in the end of the experiment because their seed coat was able to protect them. Seeing that the major problem are the seeds that remain on the surface because the seed bank will degrade fast in the conservation agriculture, we have decided to examine the influence of the crop residues on seed emergence. For this experiment, 8 wheat species were selected with 200 seeds for each trial. We used 2 types of crop residues, maize and wheat, with 3 residue quantities. 1, the quantity of residues that was produced in 1 meter square of field, half of that quantity and half more of that quantity plus control. The experiments were conducted for 2 consecutive years. Here you can see how the process was done. Firstly, the first 5 centimeters of the soil were removed and heat sterilized. Then they were put back in their respective holes. The seeds were sown on the surface and covered with residues or left uncovered in the case of control. Another test showed significant results for the factor species, residue quantities, and the interaction between the species and the years. As shown here, quantity normally produced in the field and the quantity greater than the one produced in the field influence the emergence negatively. On the other hand, lesser quantities can actually promote the emergence, which is something to consider if the crop residues are used as mulches. Single species had a diverse response to residue covers. Overall results show that the emergence of Sorghum halepense is promoted by the residues. Amaranthus retroflexus seemed to have little to no influence, while the emergence of all others is actually diminished, especially for Chenopodium album. Considering the interaction between the years and the species, we can clearly see that the meteorological situation in the 2 years was very diverse, which reflects on the species emergence. We can see that for Sorghum halepense, the differences are low between years and that the emergence is always promoted. There are also no changes for Amaranthus retroflexus, but we can see interesting results and completely opposed for Abutilon theophrasti and Setaria pumila, that actually confirm us that the current models should be recalibrated for their use in conservation agriculture. Finally, in order to fully understand the reach...
just 2 minutes left.
Okay, thank you. To fully understand the reach and the lack of our current AlertInf model that was made for conventional agriculture, we have tried to use it to simulate the data of the no-till simply by changing the temperature to the one of the surface and also using the rainfall. Here you can see the example of Abutilon theophrasti without residues. And it can be seen that the simulations are quite good, especially for identifying the 80% of emergence, which is the control threshold. But if we do the same with wheat residues or maize residues, we can see that the simulation is not good. In this specific case, the model anticipates the emergence for about a week in 2019 and for almost a month in 2020. With other species, the situation is different. The model sometimes anticipates or postpones the emergence. The motive behind these errors is the influence of residue cover on the soil temperature and humidity, and by also being a physical barrier. In order to have good simulations, the correct temperature and humidity must be used, which are the temperature and the humidity below the residues. And there are some models that can actually do this. But if the problem is the physical barrier of the residues, we actually do not have models that could actually deal with this. Therefore, it is important to collect more data about the behavior of different species and under different residue types in order to be able to calibrate our models correctly. To conclude, it was shown that prescription maps can be a valuable tool for precision weed control, that combining time-specific and site-specific methods, weed control can be more precise with a major herbicide use reduction. We have shown that integrated measures must be cautiously applied in order to avoid negative results, and that the application of the aforementioned tools is possible in different agricultural systems with a prior calibration. As 3 years are not enough to conclude such a work, I would like to consider that this is only the first step and that in the future we'll be able to move from ideas to action. Much of the work presented here is theoretical or modelistic. Therefore, real field trials are necessary to completely understand whether our theories are completely accurate. In addition, much of the work considers only a local scale. Therefore, it is important to conduct these experiments on a much larger scale in order to be able to cover different scenarios and offer the best possible solutions. I thank you for your attention, and I leave you with a bit of self-promotion. Thank you.
Thank you, Nebo. Questions?
Just some point. I can ask Nebiosa? Yes, thank you. I was wondering why cellulolytic microorganisms, if I understand well, are more...
Sorry, I just...
maybe with the slide, I don't know. I was wondering why cellulolytic microorganisms are more abundant in no-till field with respect to buffer or undisturbed pristine zone.
Yes, actually, we haven't conducted a surveys on which species exactly were present, considering the microorganisms. But our idea was that this activity is actually linked to the soil structure, and that in the no-till field there were more pores that have allowed for the aerobic microorganisms to develop and to attack the seeds, which also let's say is in confirmation with the meteorological data from the periods in which the trials with the fertimeters were conducted. Another possible solution is that in the buffer strip, there was much more cellulolytic material, considering that it is a space between the two tree rows. Therefore, it is filled with the tree litter. Therefore, it is possible that the microorganisms were already had enough of cellulolytic microorganisms, cellulolytic material. Therefore, they were not that prone to colonize also the seeds that we planted there.
Just other question. Under a certain level of quantity of crop residues, weeds emerge better than without. No?
Yes.
How can be explained?
Yes. Of course, firstly, it depends on the weed species. But considering that in the particular case of conservation agriculture, where we have seeds on the soil surface, we can see that they are much more exposed to the environmental factors. Therefore, if we have a small layer of residues on top of them, this small layer can actually reduce the temperature that these seeds are exposed to, and can also contain more moisture respect to the seeds that are left uncovered. Therefore, this can actually promote the emergence of some species in this case.
Okay, thank you. I don't know if there are other questions.
Well, maybe just a short question and comment related to the previous one. The effect of these residues on the soil surface, and you actually, in your presentation, you brought up the example of abutilon, which was like its emergence was very much delayed compared like using this alert-inf model, like for the emergence. Could you, because it's a kind of weed which has a relatively like prolonged period of emergence and everything, and it was still delayed by this. Can you give us more like a rational or reasons why do you think it's so? Like,
yes, well.
So because the first point, it's very important that you said that if somebody is using this alert-inf model, it should be actually calibrated for if there is like a cover like crop residues or any like plant residues, and that's very important, you know, like yes, conditions, yes. Not just temperature and water. It's also thank you.
Yes, well, as I previously said, the alert-inf model, as it's now, is actually only calibrated for the conventional agriculture. Therefore, when the seeds are inside the seed bank. Therefore, we have actually tried to see if it is possible for the model to do the simulations when the seeds are on the soil surface or are covered with the crop residues. And of course, we can see that the crop residues create different conditions for seed respect to the soil. Therefore, this is why the alert-inf was not actually able to do good simulations. Therefore, we need to integrate.
So what do you think when somebody is predicting like weed emergence with alert-inf, because it also comprises maybe some of the seeds are left on the surface, you know, like even in the conventional, some of the seeds could be left on the surface. And if there's some like, even in conventional agriculture, that should be taken into account that if there are some like cover, like crop residues or some plant residues, if in the case, of course, if the soil, if the seeds are left on the surface of the soil.
Yes. Well, if they are only left on the surface without any crop residues, we can see that the alert inf can actually make a good prediction.
So,
but of course, yes, sorry.
No, you could just go on.
Yes, but of course, if we have crop residues, we will have to recalibrate it. We were also actually surprised to see that the emergence of abutylon was this much influenced by the crop residues. Our conclusion was that it actually depends on the meteorological data, because the residues, if well compacted, can actually completely reduce the infiltration of the water, and can also reduce the temperature very much. Therefore, the seeds of abutylon cannot accumulate enough moisture and temperature in order to start their germination, because the seeds of abutylon are actually quite big, and we have found the references that they actually need quite a lot of moisture and temperature in order to start their germination.
Thank you. Thank you very much.
You're welcome.
Okay, no other questions. We can move to the next one. Thank you, Nebo. Thank you very much.
Thank you.
A pleasure. Thank you again. And the next one, Alessandro, if you want to introduce.
Yes, is Alessandro Pellizzaro. That okay, I leave you to present your topic, the title, without that I repeat.
Where is Alessandro?
Okay, here I am.
Ah, okay.
Good afternoon.
Okay, you can share and do your presentation. You have 20 minutes. Sometimes I don't interrupt you with telling you that 5 minutes are left, but because I'm following your presentation, so that's all. You have 20 minutes. You can go.
Okay, thank you. Thank you, Professor Bouton. Good afternoon to everyone. My name is Alessandro Pellizzaro. I'm a biologist, and I have been working in Acqua del Campo laboratory since 2004. Acqua del Campo is the in-house manager of the integrated water service for 10 municipalities in the province of Vicenza. I'm very pleased to introduce my PhD project results. The aim of the study is the evaluation of uptake and translocation of perfluoroalkyl acids and chromium by hydroponic crops and constructed wetlands. The supervisor of my PhD project is Professor Borin, and the co-supervisor, Professor Del Ferro. The project has been developed in collaboration with Acqua del Campo. Today, we are going to focus on the perfluoroalkyl acids results. In the past 8 years, high levels of FAS have been detected in underground, surface, and wastewater in the southwestern part of the province of Vicenza. One of the sources of the contamination is a fluorochemical plant area that manufactured FAS since late 60s. And further FAS sources are wastewater treatment plants that collect waters from industrial districts to a single pipeline represented in the picture by the green line. Then the big pipe discharges in the river Frattagorzone, which is an important irrigation waterway. Perfluoroalkyl acids are characterized by a strong carbon-fluorine bond that enhances the stability of perfluorinated chain and by an amphiphilic nature. Because of these characteristics, FAS have been widely used to produce emulsifying and tension-active products for cleaning and water-resistant products or tissues like Teflon and Gore-Tex. They are endocrine disruptors, and in 2009, FOS was listed under Annex B of the Stockholm Convention on Persistent Organic Pollutants. In 2017, the International Agency for Research on Cancer has classified FOA as possibly carcinogenic. FAS are usually classified in perfluorocarboxylic acids and perfluorosulfonic acids, depending on the functional group, carboxylic or sulfonic. We studied 14 perfluoroalkyl acids, 11 perfluorocarboxylic acids with 3 to 13 fluorinated carbons, and 3 perfluorosulfonic acids with 4 to 8 fluorinated carbons. The objectives of the study were the evaluation of FAS model uptake by different hydroponic crops, the assessment of FAS uptake by aquatic macrophytes in order to estimate the human exposure through the food chain, in order to assess wastewater phytoremediation by selected plants. 2 experiments were performed in an unheated greenhouse located in the Arzignano wastewater treatment plant area. Hydroponics by horticultural crops, and constructed wetlands mesocosm by emerged aquatic macrophytes. Plants were exposed to FAS spiked solutions and wastewaters. All waters were added of nutrients. And at the end of the exposure period, we determined the FAS concentration in roots and leaves and bioaccumulation factors, root concentration factor, leaf concentration factor, and translocation factor. In order to determine FAS concentration in vegetal biomasses, at the end of the treatment period, plants were divided in shoots and roots and dried in the oven. Dried biomass samples were weighed and treated with methanol. The extracts were concentrated and passed through cleanup cartridges before the analysis in a triple quadruple mass spectrometer. After the evaluation of FAS concentration in roots and leaves and vegetation dried biomass, plant uptake and translocation of FAS were determined according to different bioconcentration parameters for all perfluorinated compounds. Root concentration factor and leaf concentration factor were respectively calculated as the concentration of a compound in roots and concentration in leaves divided by its concentration in solution. Translocation factor was determined as the concentration in leaves divided by its concentration in roots. Hydroponic test was performed by deep flow technique in a recirculating that hosted 60 plants of lettuce and spinach. 12 plants per batch and per species were analyzed after the exposure period. And in each batch, lettuce and spinach were fertigated by the same solution. Hydroponic test was performed by 2 sessions. Synthetic FAS mix solution, hereafter called Lab 500 and Lab 5000, and 3 wastewater treatment plant effluents were tested. And in this table are reported all perfluoroalkyl acids concentration. This bar graph shows FAS concentration determined in roots and leaves of crops treated by Lab 500 solution. We can observe that most of FAS concentration in roots were higher than leaves for both crops, and most of fast concentration in roots, in lettuce biomasses were higher than spinach. These histograms represent root concentration factor in logarithmic values of each compound in both crops treated by Lab 500 solution. We can appreciate the sigmoidal pattern of perfluorocarboxylic acids root concentration factor and the increasing of perfluorosulfonic acid root concentration factor with the increase of increasing of perfluorinated chain length. This graph shows translocation factor from roots to leaves of both crops treated by Lab 500 solution. We can observe that the translocation factor decreased with increasing perfluorinated chain length. Furthermore, translocation factor of spinach resulted, represented by the blue bars, resulted always higher than lettuce, represented by the orange bars. These two graphs report perfluorocarboxylic acids root concentration factor values in all water tested in a hydroponic experiment. We can observe that root concentration factor of perfluorocarboxylic acids in wastewater treatment plant effluents showed the same pattern of lab solutions of perfluorocarboxylic acids root concentration factor values were similar among the test sessions. This is very interesting because similar root concentration factor data were determined by different water treatments, different crops batches, and different climatic conditions. Similarly, we can observe the same pattern and similar values between root concentration factor of perfluorosulfonic acids in all waters among the test sessions. Calculating the total mass of fast uptake by both crops, lettuce uptake with 15% of total dosed mass was higher than spinach with 4%, and estimating a daily intake of 6 grams of dry weight of lettuce for a person of 70 kilograms of body weight, only lettuce treated by perfluoroalkyl acids spiked solutions showed greater values than tolerable in the daily intake of 0.63 nanograms per kilograms of body weight for the sum of these 4 FAS, according to the to EFSA guidelines. Summarizing hydroponic test results, root concentration factor of perfluorocarboxylic acids showed a sigmoidal relationship with the perfluorinated chain length. Perfluorosulfonic acids root concentration factor increased with increasing perfluorinated chain length. Translocation factor value for all compounds was higher in spinach than lettuce, and lettuce uptake was higher than spinach. You can find further data and details of the first hydroponic test in this paper published in the Science on the Total Environment. The mesocosm experiment of constructed wetlands was performed last year from February to July, and it provided measurements of fast uptake in free aquatic macrophytes, Iris pseudochorus, Phragmites australis, and Tifa latifolia. The mesocosm was constituted of 4 batches. Each batch represented a specific contaminated solution treatment, 3 fast spiked solutions at different concentration in nanograms per liter, and 1 wastewater treatment plant effluent. In all batches, there were 3 replicates for species and 4 plants per tank. all tanks were filled by 6 kilograms of expanded clay grains. This table shows the nominal concentration of fast spiked solutions, hereafter called Phyto 500, 2500, and Phyto 5000, and the fast concentration in the wastewater treatment plant effluent. This bar graph shows the fast root concentration of each halophyte after Phyto 5000 treatment period. We can clearly observe that fast concentration in Iris is generally greater than Phragmites and Tifa. Comparing root concentration factor data of wastewater treatment plant effluent of Arzignano and fast spiked solution, Phyto 500 solution, we can note a similar pattern and similar values. In this, examining Iris flower compartments treated by Phyto 2500 solution, we can appreciate the decreasing of fast concentration by this order, from stalk to flower, from flower to capsule, and from capsule to seeds. Furthermore, fast concentration in all compartments decrease with the increasing of perfluorinated chain length. Considering the plant's response to the treatment, total dry weight of halophytes didn't decrease with the increase of fast concentration. This table summarizes principal data for the fast mass balance at the end of the test period. We can note that the absolute value of the difference between the theoretical mass of fast dosed and the experimental calculated mass was lower than 40%. This bar chart shows the percentage of fast removal efficiency of aquatic macrophytes. In particular, the percentage ratio of perfluoroalkyl acids accumulated in leaves and roots of each halophyte. We can note that the removal efficiency grows with increasing fast concentration for every macrophyte, and the Phragmites australis showed the greatest value, reaching the 81%. Moreover, Phragmites accumulated in roots up to 88% of the total fast removed. Then, observing the percentage removal of each perfluoroalkyl acid, we can note that Iris, represented by yellow bars, mainly removed all perfluorosulfonic acids and the perfluorocarboxylic acids in this perfluorinated chain length range. Tifa, represented by the brown bars, mainly removed the shortest perfluorocarboxylic acids. Phragmites, mainly removed, represented by the green bars, mainly removed perfluorocarboxylic acids in this perfluorinated chain length range.
Alessandro, for about 4 minutes, but you're concluding, so okay.
Thank you. Starting, going to the general conclusions, fast root uptake and translocation were dependent on the perfluorinated chain length, the plant species, and the bioavailable concentration of compounds. Horticultural crops treated by selected wastewaters in a worst-case scenario showed a fast uptake such that the human daily intake is compliant with the current EFSA guidelines. And in constructed wetlands mesocons experiment, Phragmites australis showed the highest removal efficiency, reaching 81%. Starting from these interesting results, we could improve knowledge about the fast uptake and phytodepluration by constructed wetlands. Testing further matrices like sludge and leachate, testing other substrates, and we can apply a long-term exposure to verify fast reallocation from shoot to roots and the system saturation. Finally, we can evaluate the removal efficiency by other aquatic macrophyte species. I am thankful to my supervisor, Professor Borin, for the opportunity to undertake my PhD in crop science, and my co-supervisor, Professor Del Ferro, for the precious assistance and suggestions. And I am thankful also to my laboratory colleagues for the support and the collaboration, and to Acqua del Campo for this great experience and professional opportunity. If you have any questions, comments, or suggestions for further investigations, you are welcome. Thanks for your attention.
I'm curious about one aspect. In the case you use phytoremediation to remove these contaminants from soil, from water, okay? And how do you treat the plants once you want to remove them, and to remove also the contaminants within these plants? There is any clear way to treat this plant and eventually plant new ones to remove again more contaminants, or?
Sorry, I lost some words in the first part of your questions.
No, I mean,
sorry.
It's just a curiosity. I'm not an expert in the field, so I'm just curious about the practical. How do you practically remove the plants that are that accumulate these contaminants, taking it from the soil, okay, from the water, okay, and you want to remove the plants to remove definitely the contaminants, right? Once they phytoremediate the soil and so on, and the water, you want to remove the plants to remove the contaminants, maybe, or not?
Okay. Thank you for your question. This is a good question, and this is the future of the applications in this field, because the problem is the treatment of the vegetal biomasses with these values of fast concentration.
Yeah, this is my question. How is there a way to treat these biomasses, or at the moment, or no?
No.
Okay.
In this moment, we have to think how to treat biomasses, vegetal biomasses.
Okay, thank you, Alessandro.
Thank you for your question. You're welcome.
I have just a technical question. I hope that about root bioconcentration of perfluoroalkylic acid in lettuce seems greater than spinach. It's correct?
I'm going back to...
I mean, root bioconcentration of perfluoroalkylic acid in lettuce is greater than spinach. It's okay?
Yeah.
It's correct?
In this
in root, in the slide 13.
In the previous one.
Okay. This is the fast concentration.
Okay.
And in this bar graph, we can note that the concentration in the roots of lettuce are greater than spinach.
There is a possible explanation for this.
In general, some authors in previous studies think that there are affinities, particular affinities between perfluoroalkyl acids and macromolecules like proteins, and different amount of proteins in different crops can give different results of the uptake and then of the root concentration uptake. Then more hydrophilic compounds like PFBA can better translocate from roots to leaves to shoot.
Okay, in this case also the sigmoidal pattern of this perfluoroalkylic acid, probably at the if you come back to the next this slide, probably the sigmoidal pattern means that the first compounds FAS are uptake at the higher level, then decrease. What is the reason why this short molecule, because are short in chain, right?
In this case, some authors in previous studies think that the root concentration factor of perfluorocarboxylic acids, depending on the perfluorinated chain length.
But this chain length is short or not?
Yeah, but there is also the pH, the pH value in solution.
Okay.
That cause different different uptake from solution to the roots, because we have to consider the dissociated form of perfluoroalkyl acids and the undissociated forms of perfluorinated compounds. And give an example, at neutral pH in solution, we can have perfluorobutanoic acid in dissociated form, but this form is not this form is not able to cross the membrane than the undissociated form. The undissociated form is more hydrophobic than dissociated form. And this equilibrium, depending on the pH value, can cause this form of sigmoidal pattern.
Okay, thank you.
You're welcome.
Thank you. Yeah, thank you very much. Just a short one, just in continuation to the previous. Can you go to the previous slide, slide 13, like you said, root concentration, and this difference in between two species, you know, you used in your research, lettuce and spinach, and you said that there's some literature like they have a different combining ability to proteins. I just wonder, when you are like harvesting for these roots, are there much morphological differences between the root biomass between these two crops at the time when you harvested? Is it very similar, comparable, like root biomass when you harvested and analyzed for concentration, spinach and lettuce? Did I make my question clear?
The biomass of the two crops are
yes.
in dry weight are similar.
Similar, okay.
Very similar. Only the edible part was different.
Okay, but the root biomass was similar. Okay, thank you.
Okay, I think that we can move to the next. Any other question? Thank you, Alessandro. Yeah, please share. Okay, thank you very much.
Thank you.
Alessandro Raiola, do you want to introduce the next one, or do I have to do it? I can. Okay, the next
is Stefania Sut. Then will present us the topic that depicting change of secondary metabolites in selected crop queens and ancient apple fruits variety and grape wine after silicone application.
Stefania, can you talk?
Good morning.
Your volume is very low. The volume of your microphone is very low.
Okay. If I speak, okay, perfect.
Better in this way.
I share my screen. Good morning, everybody. I am Stefania, and I would like to thank the commission of today and all the board of the PhD. I would like to thank Mario Malagoli, my supervisor. The title of my presentation is Depicting Changes of Secondary Metabolites, Queens and Apple of Different Varieties, and Grape After Silicone Application. Secondary metabolites in plants are natural compounds with a complex chemical structure. And there is many literature that report that secondary metabolites have significant role in many biochemical processes that can occur in humans, animals, and plants. The aim of my thesis is to enhance the knowledge in the field of the secondary metabolites in edible plants. In particular, I divided my thesis into parts, apple and quince, and grapevine. I focus in the analysis of plant tissues. Plant tissues are complex mixture, and one of the main techniques that are used for the study of these compounds are liquid chromatography. We can perform different types of liquid chromatography based on different types of separation, and we can use different types of detectors. In particular, UV visible detector, diode array detector, light scattering detector, or mass spectrometry with different type of analyzer, like ion trap or, for example, triple quadruple. Secondary metabolites analysis can have different issues. In particular, we can have compounds that are characterized by chemical instability, for example, anthocyanins. We can have compounds that are present in really low amount, like some phenolics, and there are compounds like sugars that have no absorbance in UV visible spectra. Other compounds like triterpene acid can have problems in the ionization by mass spectrometry. So during my PhD, I developed different analytical methods for analyzing different types of secondary metabolites. And every time we have to consider the peculiarity of each class of this compound. So we prepare sample, perform the analysis, elaborate the data, and perform the identification of the compound and then the quantification. In humans, secondary metabolites of plants can be assumed with diet. and the secondary metabolites in food have health-promoting properties. There are some products present in the market that are called food supplement that are made from plant extract that are really rich of high rich of secondary metabolites. We focus on apple and quince, called in Italy melacotonia. The aim of the first part of the PhD is to assess the presence of valuable secondary metabolites in apple and quince, and to improve the methodology in this field of research. I divided this part of the thesis in 3 chapters. In the first work, we focus on the study of quince fruit as extraction source of antioxidant phytoconstituent for food, nutraceutical and functional food application. In this work, we perform the comparison between apple and quince. In the second work, we studied the fragmentation pattern of triterpene acid by LC-APCI-MS. So we developed a new methodology of analysis. In the last work, we focus on the study of ancient apple of Friuli Venezia Giulia as nutraceutical ingredient, and we apply the new methodology that we developed. In the first work, we focus in the study of the phytochemical composition of quince fruit and to compare it with those of 6 commercial apple variety. So we develop a LC-DAD-MS method that allow us to identify 42 secondary metabolites. Then we perform the quantification of metabolites and perform a preliminary antioxidant assay as DPPH assay. Here I report some results. We can see that quince extract have a higher amount of secondary metabolite compared to the other apple varieties that are, and we can correlate this higher amount of secondary metabolite with a higher antioxidant capacity. In conclusion, the quince fruit showed a quite different phytochemical profile compared with the 6 apple varieties. Quince can be valuable in nutraceutical considering the higher content in phenolic compounds and the stronger antioxidant capacity compared to the other considered fruits. So we decided to go on with the study of the secondary metabolite in apple, and we divided these classes in flavonoid, chalcones, procyanidins, and triterpene acid. These compounds have some peculiarity. In particular, triterpene acid have poor UV absorption. There are some methods for the analysis of this compound and are usually done by LC light scattering and LC-MS. These compounds are interesting because they are active because they have anti-inflammatory properties. There is scarce information about their amount in the apple tissue. So we decided to develop an LC-APCI method for the analysis of triterpene acid. And this new methodology was published in this paper in Journal Mass Spectrometry. So we decided to apply this new methodology in a real sample, and we decided to study ancient apple of Friuli Venezia Giulia. And we compare this sample with the commercial apple variety. In the experimental work that we performed was focused in the identification of secondary metabolite by LC-MS and in the quantification of the compound. Here report one of the more interesting the results related to triterpene acid. We can observe that the amount of triterpene acid was higher in the ancient apple compared to the commercial one, And with this new methodology, we are able to identify and quantify each triterpene acid derivatives. So at the end, in the first work, we performed the comparison between apple and quince, and we highlighted that quince is a good source of polyphenols with high antioxidant activity. So we have tried to understand if there is a possible uses of this fruit for nutraceutical. In the second work, we developed new methodology for the analysis of triterpene acid. This methodology can be applied to different plant materials. In the last work, we applied this new methodology and we focus in the re-evaluation of ancient cultivar of apple. Then we pass to the second part of my PhD. Plants' content in secondary metabolites can be influenced by different stimuli. In particular, we can have biotic stresses, abiotic stresses, or specific treatments. All of these stimuli can induce changes in secondary metabolite content. We focus on a specific treatment, silicone treatment that is supposed to alleviate the water plant status and improve water balance. In particular, silicone is an environment-friendly treatment that is used in biodynamic practice as 501 preparation. We focus on a specific plant, Vitis vinifera plant, that have a great economic impact in wine market. So the aim of the second part of my PhD is the evaluation of silicone treatment on plants of Vitis vinifera. We perform 3 different experiments. In the first experiment, we want to measure the effect of foliar application of silicone on the resistance to drought stress of young Sauvignon Blanc plant. This experiment was carried out in greenhouse. The second experiment want to study the metabolite changes in Garganelka leaves and berries after 501 biodynamic treatment. In the last work, we perform the grape comparison changes induced by biodynamic management of 4 cultivars of Vitis vinifera by targeted metabolomic. So the last 2 work was performed in open field, and we applied different metabolomic analysis. In the first work, the aim of the study is to investigate the effect of foliar application of silicone in Vitis vinifera cultivar Sauvignon Blanc plants under water stress. We divided the plant in 4 groups and we measure leaf area and shoot length, chlorophyll content, and photosynthesis parameter. We performed the silicone application before the start of water stress period. Here I report some results. In the graph we can show the total shoot length and the total leaf area. In water stress plant, silicone application enhanced growth compared to water stressed plant. And comparing the 2 graphs, we can observe that we have a similar trend. In the graph above, we can see the leaf chlorophyll content. We measure chlorophyll in 3 different time points. At the light drought, severe drought, and recovery time. Plants subjected to water limitation and treated with silicone keep the chlorophyll content at the level of the irrigated plants. In conclusion, silicone partially alleviated the water stress, attenuating the vegetative growth reduction. Now we passed into the discussion of the second and in the third work. In particular, we work in this case in open field, and we apply a metabolomic analysis. But we apply two different types of metabolomic analysis. In the second work, we apply a non-target metabolomic analysis, and in the third work, a target metabolomic analysis. What are the differences? In the untargeted approach, we perform the experiment, we do the extraction and the detection, and later we perform the annotation of metabolites and statistical elaboration, and then the interpretation of the results. In the targeted approach, we perform the identification of the metabolites of interest. Then we do the calibration of the mass detection, and then we perform the analysis in the extract, and then we perform the interpretation. In the second experimental work, we work in open field in Garganega cultivar. We want to assess possible metabolomic changes induced by the application of preparation 501. We analyze buried tissue and consider the element content, the amino acid, and we perform untargeted metabolomic analysis. Here we report some results. In particular, in the graph, we can observe that the blue dots are the treated group and the green dots are the control group. We can observe that we have a partial clusterization between control and treated group. Here in the table, we report the metabolites that are more descriptive for the treated group. We can say in conclusion that the application of 501 treatment induces synthesis of phenolic metabolites. In the third experimental work, we work in open field and we choose 4 cultivars: Chardonnay, Oseletta, Teroldego, and Garganega. We want to assess possible metabolomic changes induced by the application of preparation 501. We analyze buried tissue and perform a target metabolomic analysis, and we focus on the analysis of polyphenols, anthocyanins, sugars, and amino acids, and then we perform the multivariate analysis. Here I report 4 chromatograms, 1 for each cultivar, and we can observe that each cultivar have a specific fingerprint. So we decide to elaborate separately each cultivar. Here I report as example the analysis of Oseletta cultivar, and we analyze polyphenols, anthocyanins, sugars and tartaric acid, and the amino acid. Then we merge all these data and perform the multivariate elaboration. This elaboration was performed for each cultivar. And here are the results. We can observe that comparing the 4 cultivar, only in Oseletta we can observe a clear clusterization. In particular, we can observe that the treated sample have a higher content of anthocyanins and a decreased amount of amino acid. In the other cultivar, there is a minor difference between treated and control group. But at the end of this experiment, we want to know if there is a common trend in metabolites between all the cultivars. So we want to emphasize the difference between 501 treated and control groups. So we decide to merge all the data, and we can observe from the graph that there is not a so clear clusterization between the group. Here in the right, we can observe the metabolite that describes 501 treatment sample. And in conclusion, we can say that a common trend among the 4 cultivars is observable in the increase of secondary metabolites. So at the end, in this first experiment, we observed that silicon partially alleviate the water stress, attenuating the vegetative growth reduction. And in the second and the third work, 501 treatment induce a common trend in the increase of the secondary metabolite, like polyphenols and anthocyanins in berries. So at the end, all this work was focused on apple, quince, and grapevine, and we depict in different way the changes in secondary metabolite. We develop different methodology that can be applied in many fields of plant science. Thank you for your kind attention.
Okay. Stefania, I have some question.
Yes.
Regarding the first part, okay. In the first part, I took a look at your materials and methods section. You said that the 6 commercial cultivars of apple that you used were bought in local stores, okay, in September. If you buy some of these cultivars in September, they have been harvested 1 year before, 10 months before, because, for example, Granny Smith and Fuji will be harvested later in October, November, and so on. Okay, so in your on the other hand, the quince fruit that you used were maybe just harvested, because Champion usually is harvested at the end of August, beginning of September, right?
Yeah.
So you compared fruits which were harvested 10 months before with fruits which were harvested just a few weeks before. And just to let you know that this can be a critical point, okay? And I don't know if you use the same apples in the other part with the old apple varieties. If you use the same, the critical point is also there. You compare all the apple varieties which were just harvested, as far as I understood, against apples which are harvested 10 months before. I'm talking to you this because the phenolic compounds, the antioxidant activity in the fruits after 10 months may change. There are several publications, maybe you know more than me in regarding this topic. There are several publications in literature where they show the factors affecting the way the apple fruits sometimes lose or increase also sometimes, okay, some phenolics and the antioxidant activity. But I think this is a critical point, and I would like you to say something about this point.
Thank you. So thank you for this comment and suggestion. I completely agree with you and with these comments. And I have this comment from my reviewer that corrected the PhD thesis, and we discuss about that. And at the beginning, we start with this research because we are focusing in the searching new nutraceutical ingredient. So we are focused on the content of secondary metabolite. And I think that is another point. Another thing is that my background is not an agronomy background. So I learned this new thing that I really have to put attention of the origin of and the time of the material that we are analyzing.
And the place where the fruit were grown, and a lot of other factors, the post-harvest conditions and so on.
Exactly. So I completely agree with your observation, and I keep this in mind for further works. So I think that in this point I learned so much, and I completely agree with you. Thank you.
Okay, I have another comment concerning the second part. Okay, I don't want to enter in the polemic about biodynamics and so on, which is already is always open. There are big discussion, and it's quite controversial. But I have a general impression about these problematics regarding your thesis. I think that the biodynamics practices can be, of course, you have the right to evaluate what you want, of course, okay? Many of these practices were already studied and reviewed, and many of these practices were shown to be absolutely inefficient, okay? And effects were shown to be by random sometimes, okay? Or due to other factors. In this case, you used this preparation, and I think that you can call this preparation with the right name in your thesis. It is silicic acid. It is silicic acid of which you don't know the concentration, right? And this is an important point. And I think that from your side, you said, I'm not an agronomist and so on, but from your side, I think that you should have taken this into account for several reasons. First of all, if you want to replicate an experiment, you must be sure to use a preparation with the same concentration. Okay, just this to be naive, but there are much more complex aspects, of course. And this is one thing that I would like to from you to comment. And yes, basically this. I have another thing in my mind I don't remember, but basically this. If you have a comment, yeah, of course, the effect of silicic acid on cells, on cell walls, especially, you wrote it in your thesis, okay? The effects of this silicic acid on cell walls are proven, are true effects. So, of course, in this case, there is a scientific base, but sometimes I think that the form is more important than the content, okay? And to call this preparation with the right name, I think should be the first step towards a more scientific approach. This is a general comment. I don't know, maybe you have something to say about it.
I completely agree with you again. I know that is a controversial topic, and there is many literature that study biodynamic treatment, and we try to set up this experiment, and I think that we try to do our best in this point of view. I take the sample and I perform the elaboration without any prejudice, I don't know, say in English, on the treatment. I label the sample like control and treatment. I don't put so much attention on what is the treatment, and I only observe the elaboration data. So
yes, Stefania, but let me say something. When you have a scientific approach on a problem, you must follow the scientific method, okay? And I think you must. I think we have, no, I don't want to say you must. I would like to say that you must be linked, at least, to the scientific method. You are free to do something more, but you must be linked to a scientific. This is a scientific thesis, okay? So you must keep it into your mind, of course, but I think you know it. It's not necessary that I told you this, but and so you must also, for example, make a hypothesis. You, on one hand, you, of course, you have all your control. I don't. My criticism is not towards your experiments. Okay, is more towards the general approach. Okay, so but
Okay.
it's not so critical. It's a suggestion, my impression.
Thank you so much.
Okay,
I agree
I'm
with
not,
you.
Of course, what you wrote in your thesis and which is already organized in a way that you can publish maybe small articles. Okay, and I think that you have to work a little bit more to increase the level, because the results are there, of course. But the key of lecture of these, the key that you have to use to read these results must be a little bit more higher. Okay, this is my accept this critical opinion. It's not a bad opinion.
I know that our work is borderline.
Okay,
but
thank you.
We have to try everything.
No, anyway, thank you for presenting us this work anyway.
Just in continuation to these previous comments from Professor Alessandro, Stefania, also thank you for your presentation. I just want to, what do you think, like, now I found out that your background is not agronomist and like that, but can you, if you recall, we call treatment 501, and it had some effect on berries, okay? Yeah, you presented, it had some effect. And then you jumped to like 4 different cultivars of grape wines. You understand? This is what is professor, but just listen, like this is what is professor saying, to find sometimes a difference between species, like the first one between quince and apple, it's easier. But to find difference among cultivars within the same species, it's really hard. You understand? It's difficult to expect. You understand? Even if you, for any very important treatments, even like, just to make it very simple, even nitrogen, like I don't know, it's difficult to find cultivar specific responses. So I just want to support, like it's all with good intention from the previous comments, that it's very important, and that's one of the bases for PhD work actually, that any PhD student to become independent in scientific thoughts. You should learn to work independently now, independently to everything, to set up a new research and now independently, with the help, of course, of the previous all documentation and literature. So that was the only, you understand, like difference that was not expected, and I was like surprised. Okay, thank you very much, Stefania. Oh, wish you all the very best.
For your suggestion.
For this small comment, just in continuation to always to the previous comments. Thank you. So that's all.
Okay. I don't know if there is just one curiosity. No, you hear me?
Yes.
Okay. No, just curiosity, probably previous was close my microphone. If you have tested in field for grape,
yes,
no? White and black berry. So 501 preparation seem affect the metabolites differently between white and red grape. In particular, you have identified or detected some marker that distinguish white in this cultivar that you have evaluated. There's some marker metabolite that can be present only in white, but not in blackberry. There is something that.
So when as I show in this slide, each cultivar have a peculiar pattern of secondary metabolites. And in particular, teroldego and oseleta that are blackberry, we have the quantification of the red pigment anthocyanins.
But
so
In protein, there is some peculiar amino acid that...
No, but we analyze all the essential amino acid.
Okay.
And we obviously analyze anthocyanins in the red berries, and we don't have in white berries. So we analyze other type of compound. And this work was a challenge because we try to merge all the data, even if the cultivars are very, very difficult to each other. So we decide, for this reason, we decided to elaborate each cultivar separately at the beginning. And then we try to stress the data for try to see something, but we know that there is this limit due to the presence of 4 different cultivars.
Thank you.
Thank you. You're welcome.
Okay, the section is completed. Alessandro, you have to open the microphone.
Sorry, guys. Now we are going to meet, the commission is going to meet separately into a breakout room. And we have to tell the students when we are going back for the proclamation. I don't know if Zlatko and Alessandro, do you have an idea? Half an hour for us is enough for the judgments?
Before to meet again?
Yeah, before meeting again publicly.
20 minutes, half enough. Zlatko,
yeah, I'm okay. 20 minutes is also fine with me.
Okay, so we can be back at 1:45.
Yeah, that's okay.
Okay. So I will put, I will ask a colleague of mine to put a slide with the time we will be back. Okay. So I create the breakout room, and
yes, we enter.
We go there, and allora.
So we just stay connected and we will...
And I will let you know when you will be added to the room.
Is in a breakout room.
Sorry, 1 and 2. Yes. No. This is not a sign. Okay, we are back. We had a long discussion. Now also the president will come back and will read the proclamation. Okay. So sorry for the delay, but we wanted to be sure to give a well done comparative evaluation of your work. Okay. And we hope we did our best, I think. Alessandro is back, so Alessandro, you can start when you want to read the proclamation. We cannot hear you, Alessandro. Turn on the sound.
Sorry. You hear me?
Yes.
Okay. I start with the proclamation of a PhD. First with the Dr. Monica Canton. Where is Monica? Okay. The committee unanimously judges the work done by the candidate Monica Canton as excellent, approves the thesis and proposes to the Magnifico Rettore to issue the title of Dottore di Ricerca.
Congratulations, Monica.
Thank you very much.
Dr. Manuel Ferrari. The committee unanimously judges the work done by the candidate Manuel Ferrari as very good, approves the thesis and proposes to the Magnifico Rettore to issue the title of Dottore di Ricerca.
Thank you to all of you.
Congratulations.
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 Dr. Rashida Govind, the committee unanimously judges the work done by the candidate, Dr. Rashida Govind, as good, approves the thesis and proposes to the Magnifico Rettore to issue the title of Dottore di Ricerca.
Thank you very much.
Congratulations.
Congratulations. Dr. Matteo Longo. The committee unanimously judges the work done by the candidate Matteo Longo as excellent, approves the thesis and proposes to the Magnifico Rettore to issue the title of Dottore di Ricerca. Congratulations.
Congratulations.
Congratulations, Matteo.
Dr. Nebbiosa Nikolic. The committee unanimously judges the work done by the candidate Nebbiosa Nikolic as excellent, approves the thesis and proposes to the Magnifico Rettore to issue the title of Dottore di Ricerca. Moreover, the committee proposes to issue also the Doctor European label to Dr. Nebbiosa Nikolic. Congratulations.
Congratulations.
Dr. Alessandro Pellizzaro, the committee unanimously judges the work done by the candidate Alessandro Pellizzari as very good, approves the thesis and proposes to the Magnifico Rettore to issue the title of Dottore di Ricerca.
Congratulations.
Congratulations.
PhD board, thank you.
Congratulations. Dr. Stefania Sut, the committee unanimously judges the work done by the candidate Stefania Sut as good, approves the thesis and proposes to the Magnifico Rettore to issue the title of Dottore di Ricerca. Congratulations.
Congratulations, Stefania.
Everyone.
Yeah, congratulations to everyone. You did all an excellent work. But of course, we have to decide a comparison and to make a comparison, and that's why you find out different adjectives in your final proclamation. I hope you are satisfied, but I think that the most important thing for you now is to look at the future. The future is yours. So good luck with my heart. Okay.
Thank you.
Thank you.
And I think we can conclude. Just the 3 members of the commission can remain. You can go and drink a lot, I hope, respecting the rules, of course, but you can drink alone eventually.
I just want to say, have a good day.
Yeah.
Thank you.
Thank you. Thank you very much.
Okay.
And so please disconnect and bye. Ciao, ciao.
Bye.
Bye.
Bye. Bye.
Bye.
Bye bye. Ok, ci sono altri che sono qua dentro, ma vediamo se riesco a buttarli fuori.
Chi sono i 6?
Rimuovi.
Beh, Claudio
può restare, ma questi qua...
come si fanno a buttare fuori?
Sto cercando di... I'm trying to put these people out except for Claudio, but I'm not able to... we can go in a breakout room and it's okay. We can go there. Yeah, okay.
Sì, sì.
Again. And so we can enter again.
Se no gli si può mandare un chat.
No, vabbè Alessandro, che restino là pure.
Sì, sì, ok. Hai ragione.
We go to the Sala 1.
Ok.
I'm waiting for the link, yeah.
Where is to enter in break room? Sala gruppi, enter.